wish I had time to put this in a PowerPoint, but I don't. So here's the first image. Who is it first?
Gene Myers.
Gene. Okay, nice. And then what's the context?
Sweatbox 2008.
Nope. He was trying out for the Backstreet Boys. No, actually, Simran, I mean, I'm going to show a bunch of pictures, and I was not here for any of them, so I don't know the context. This wasn't the music. This wasn't the audio.
I'm looking for the year. Just give me a second.
Because it really does seem like that might
Yeah.
is
In
also.
2003, this was the party in which we celebrated
That looks like singing to
the end
me.
of Savara, and we had in my basement performance.
That in our basement.
Yes,
That might be
it was in Maryland. Yes, in our basement. We have a musical director who is not here there. And here, Marissa is supposed to come here today. And
Was Marissa who I just showed?
Marissa was the singer. Jason was the big musical man. Jason, who else?
Russell Turner.
Russell Turner was the director. He's not coming. Unfortunately, Hagit passed away, one of our dear colleagues. She was doing clarinet.
Hold on, let me make this bigger.
Here is the
Oliver?
musical director.
Oliver was playing too.
Yeah, Oliver too. He was playing organ.
Russell Turner.
This is playing guitar. It just happened to coincide with my 50th birthday.
That's Hagit.
This is Hagit.
That's
Flute, right? Ian.
Ian.
I don't think there are any. I'm just checking to see if there are any embarrassing pictures before I flip the slides.
Here is Benit was at the, remember when we were in my basement? Oh, this is another one. Russell was there.
That's a fake bird.
That's a fake one.
Yeah, it was October, so he came dressed up Halloween costume. Good job, good job.
Wait, what's the costume?
Pirate.
Oh, pirate. Okay. Russell, you have to have a patch over your eye or something.
What is your
Oh, I think Russell decided to Halloween celebration early.
Okay. I also apologize. So you can see the file name. So the file name has all kinds of suffixes on it because as a group, as a committee, we all went through the effort to digitally enhance all of these photos using AI. So there might be some weird imperfections. If you see something weird, that's totally an artifact of the AI.
We are not real there, this is fake.
This is not entirely real. No, all of these images are real. They're just upscaled, basically. So
Embellished.
yeah, a little bit. People look maybe a little bit nicer than the otherwise.
Here is Benit, you're here. Gina, Ross, you're here.
Oh, I see.
And Gene,
It was a sing-along. We had lyrics.
the sing-along, yes.
Another one with Gene.
Oh my god.
You are stealing
Like,
from my.
Oh, am I stealing? I don't want to.
No, just kidding.
Do you have old black and white photos in your?
No, that's all I have. But look, this is the
No,
surviving.
you can talk about that. You can talk about that later. I'm not
Here
gonna.
is
No.
Jason.
Okay, you don't have these.
Oh wow.
Okay.
This is from Romania.
I think this is one of. Oh, there are pictures that you can collect in the back over there where the name cards are. You can pick your favorite picture. This is one of them. This is sophisticated Soren.
PhD admission or something.
Why weren't you using LaTeX?
was before you were born. This was my office. First location.
Yeah, I'll go through these if you want me to stop.
No, that's fine.
Let me know.
Fine. This is as perfect as
This one needs to be restored. This is your class? Is
This
this
was
the,
the computing center, and Professor Ignat is the one who's moustache behind me. He's a head, like,
did you really need the hat with how much hair you have?
well, it's snow, don't you see?
That's true, yeah. What was the temperature in Romania at this,
Snow is serious in Romania.
Well, it was comparable to,
comparable to here? What's that?
You got this.
Oh, she's using your head as a, to warm her hands maybe? I don't know. More class pictures. Yeah, I think this is party story. If at any point you want me to stop.
No, and the gentleman to the left is not with the products, which he wasn't.
Also, feel free to tell me to.
I will, don't worry.
Oh, we're getting to color now?
This is Alan Cowan, the guy at the very right. So this is the inventor of class index class P. He was at Princeton, was my colleague. You could see Steve Cook, the third from the left, the guy in the back. It's famous Steve Cook. Those are Cornell people. That's it.
that's what I
Here is Steve Cook.
Oh yeah, there we go. The date.
This is Alan Cowan. Not a spectacular position.
I see you treat your guests with a lot of hospitality. Let's go to the woods.
He brought to me the Universal Sequence.
Ah, I see.
Tell me, right.
Maybe not the best. Whoa. Uh-oh. A little scandalous.
That's from a different place. I think it's from Israel. Was an iCow. I don't know how you find it, but look, I'm there in a corner.
Oh, that's you. Yeah, this is his Rocky stage.
Oh, thank you. Here is Alan to the left. I think introducing. This is Hartmanis, Juris Hartmanis. He passed away recently.
Yeah, these are in no particular order. I think they're just in order of the numbering from what you sent me. This is a hand.
Ellen Cowan, yeah.
guess we spent.
Yeah. New game. Okay, here we go.
This is a weird picture. I was eating something, I don't know. Thank you, Derek.
Hey, this is
It's
improvisation.
the entertainment portion.
You can always just say this is AI that put you in that position. Yeah, Professor Gromov is, he was connecting yesterday, I think, to practice, so. Oh, sure,
Oh, this is the best. This is the best way.
okay,
Yes, Vinit is next.
okay.
So
We'll go through these.
turn a little bit to the previous picture. But we cannot see it now.
This one?
Yeah, the gentleman in the seat.
Wait, hold on, this might be my fault.
Just so I can verify the things.
Yeah, just.
That's not my fault.
So could you
become
have AI in
This
here?
is become.
Actually, that's the in-painting problem. So you can inpaint around the picture now. People do that with like the Mona Lisa, so paint around, fill in the details.
Derek, flip a little bit fast. I want to see if we get to a point. I want to show Ernie Fred House, the director that funded us. It should be, it's one of the pictures that I'm telling you.
That is, why are these? Oh, my computer is saying it needs time. Let's see.
All right, so I think it's good. Let Benita
Okay.
go next. Oh, here is Michael Fisher and Bill Camp and stuff. Many interesting things here.
I did want to get to the Gabby. Let me just see what's next. Because we actually do have some time, and I don't think Vinny's going to want to talk for 40 minutes. Okay. Solomon Marcus?
Marcus, yes.
Is this Albuquerque?
Albuquerque.
And then we have a
Or Romanian.
more Romanian.
This is Henry Lucian, who talked yesterday. This is Sergio Rodano.
That's like, oh, I like this one.
So I was working on the P versus MP problem in Romania. You see, I believe they are different.
How did that turn out? Did you prove?
I'm still working.
If you join the shoes, they will become unequal.
Unequal, yes.
I think there's 30 photos left. Okay. There's a lot of beer. It's a bad picture. Oh, here we go. What does that say? Can you reveal?
Yeah. We had a trial for our director because he quit the director of the center, Professor Ignat, to be Dean of Science or something. So we had a trial and found him guilty of treason.
Was he convicted?
We gave him a present shaped as a DNA with all our names on it.
What's happening?
That was what this slide did.
Oh, this was the trial. Yeah.
Yeah, there are more pictures of the trial, but not here probably.
Oh, Knut?
Oh, yeah.
Lots of computer science topics.
This is the dapper Soren picture of another class picture.
That's Gabi.
Oh,
Wow.
waving his hand.
I think, I don't remember.
Festival or something, or?
It's possible that Ceausescu, the president, was supposed to dance in this one. We were supposed to chant or something.
I see. Compulsory
Yes,
chanting.
Forced love.
Oh, dancing.
Uh-oh.
Scandalous. What is this?
This is the computer center.
Oh,
The new computer on those years was
is
broken.
that a laptop?
That was like 1 megabyte.
Desktop?
Exactly. Okay, we are nearing the end here. There was one picture with Soren and Gabby together that I want to get to.
This was my car, Dacia,
Oh my gosh.
a French
The Renault.
Renault made Dacia in Romania.
This is actually one that I think the AI did pretty well on because you're totally opposite in the original picture, but it sort of cleared it up pretty well. Looks like he's saying something.
Yes. That's funny.
Okay. Sambuca or something?
Yeah.
Schnapps? Maybe that'll be storm leader. If you want me to stop, I'm just gonna go.
I think we can stop here. Let's have Vinny.
Okay. Beneath. Oh, this is the one I want to.
Whoa, nice. Gabi, remember? The early days.
I'll send it to you, Larry. Yeah, I think I'll send it to you.
So, wow. I'm gonna, we tried calling, he did not answer, we left a message. I can email John and see if he's willing to start earlier. Or,
We have a speaker now. Vinny should be next.
Yeah, but starting earlier would be
Please,
good.
please,
yeah,
please.
yeah.
It's a time that
We'll have
time
to.
found.
Yeah,
We
yeah.
have to.
Hopefully everything's okay with.
Yes.
Can
we record?
Oh, sure. Yeah.
Gabi, this is the
It's a new computer, so it shouldn't die.
I'm ready to talk.
Oh, wait.
Sorry, I can talk too, I have a short talk.
Okay, so
Oh,
let's
awesome.
do Vinny first and you too.
Okay.
Perfect, perfect. We can fill the time.
You loaded my call?
yeah.
Yep. Oh, take it off? Oh, come on, Gabby. I actually like that picture of you. Alright, we'll put Gene in there.
You can do the honorary job. I know.
Gabi, so. Oh,
Yeah, we can. I can
I
send
don't
you.
know, are you going to do?
No, my daughter said that. So it's kind of
Alright, so we.
I can come back when he shows up, or just send an email.
Yeah, we'll send you an email.
Actually, around for.
Once we get a connection, we schedule and we send.
Okay, thank you.
Sorry, Gabi.
Yeah, thank you.
That's
If you could get a copy of that, because he's in Israel.
France. No.
In France?
Yeah, he was called. Maybe he has a different time or something.
Yeah, no, it's 11:05 this schedule.
You just barely
You
got
know,
my
this
hand.
was very late.
Oh, you're. Wait. Yeah, where were you?
Somebody.
Here, that was,
Oh, there's.
Go. here is our last, the remaining 6 of the Celera group after we were laid off one by one.
There.
There you are. You play the banjo?
I do. And Karen Remington left me her banjo.
So Karen passed away and we were together.
Or Vinny, I have your slides if you wanna.
Okay, I just want to send a mail to Ron, just in case, because it's late for him in Israel when he's talking, and if he's available, he can talk now.
Oh, sure, sure.
All right, people, hopefully everybody can hear me fine.
Yes.
So, this is a very short talk. Soren has so many well-wishers that I didn't think that I would actually speak, but we've known each other for quite a while. And in 1995-96, NSF DIMAX had a special year in computational biology at a time when the field was still establishing itself, and I was a postdoc then. And I met Soren. I'd never met him before, but my colleague Ravi, who couldn't be here today, but he sends his regards. So I met Soren, and the first question he asks me is a question relating to sorting of cards. And I think people already, may have heard about the work done by many people like Percy Diaconis and others, where you have a sorted deck and you do some kind of shuffling and you get a random deck, right? And the question, of course, is how many shuffles does it need? And you can show that in about 7 shuffles, especially if you use what they call the riffle shuffle, where you break the deck up and then you shuffle them together. It takes about 7 shuffles to get something randomly sorted. But Soren asked a question that's really not a math, it's a discrete math question, one that only an algorithmicist would love. And he said, supposing you had the power to shuffle in the way that you could, all right? So you can do any kind of shuffle as long as you break up the deck of cards, you put it together in any order, as long as they maintain their current order. How many steps would it take to go from a completely random shuffle back to the sorted shuffle? And of course, what this has to do with genome rearrangements that I was working on, only Soren knows, and maybe he can say. But I went back home, I thought about it, and to my surprise, and also to Soren's, we actually had a good solution to the problem, which I don't remember. I tried to dig it up now. It never was published, but it exists somewhere in our collective minds. Right, and so that was the beginning of a great mentorship, I should say. Soren invited me to Sandia soon after. I had 3 wonderful weeks there. Gabby, Soren, we met often after work as well. And many happy memories from that time. We actually didn't publish for many years after that, almost 5-6 years. Sorry, did it go out?
Oh, shoot. Yeah, it's because it's on. It's not plugged in.
All right. Right. So we it was only back in when we met again in Celera, again through some through the gods smiling on us, that we actually got to work together on haplotype phasing, another of Soren's many phase transitions. So I think a few things that I want to say. I only have 3 slides. Is that Soren, of course, as has been mentioned many times, is really influential in the community, not so much because of the work that he did, but because of all these random encounters that he had. He would find people who he thought were doing interesting work, challenge them, and then sort of motivate them to do their best work. And you can see, sort of as you go through the photographs over the years, Soren's always there in the background, sort of spurring people on. I picked this photograph because Andy spoke yesterday, and I think you'll agree that both Soren and Andy just look as young as they did 20 years ago. And this is another interesting photograph from Poland, where the Recom steering committee was meeting, and you can see the dear leader sort of blessing us in the background. So I'm sorry? yeah. Brezhnev and some other people. All right, so if you've had the pleasure of receiving emails from Soren, as I have, many years, you will find that there are certain phrases that are really Sorenesque in there, and they include words like randomness and rigor in computational biology, lively scientific dialogue, as well as interspersed with names of his heroes. So we see von Neumann and we see Rosalind Franklin up there. All right. One last picture, because I was going to actually present just before I introduced Gene today in one of the keynote talks today. So maybe I thought I would show you a photograph from the Celera days in happy times. This was probably around the time of the celebration of the assembly of the human genome, and you can see Soren and many other colleagues in the community, and of course I think one of the pleasures from that time onwards is that both Gene and Soren did their best to recruit people, irrespective of whether those people were agreeing with them in the work that they were doing or that they were disagreeing, and they would actually take a lot of trouble to seek out people whose thoughts were counter to theirs, and then and they brought us all together. And I would say that the fellowship that was created then survives to this day. So thank you, Soren, and happy birthday. you gonna go next?
No, Ron?
All right.
Oh, okay, I'm sorry to give it.
Oh, no, he's getting it set up, so he just joined. He's getting it set up, so he's not ready.
Not ready? Okay. Yes, what?
Oh, where are you again?
Shepherd University, near Frederick, Maryland.
No,
Associate professor, yes. Yeah, you don't need to introduce.
Oh, but I want to. You deserve it. You deserve it. This is my pleasure to introduce Jason Miller, famous for playing the banjo. Oh yeah, sure. I won't do the joke again because it's awkward. It's my pleasure to introduce Jason Miller. Jason was at Solera with Soren. Now he is an associate professor at Shepherd University. And please, whenever you're ready.
Thank you very much. And we met at Brown. We had a fun
Yes.
time. I think we should be keeping track for each speaker. So I come in at four publications with Soren, 4 Sorens. Those were a wonderful couple of years that I spent with you. So thank you for pushing me to work on these papers and to publish them. What I want to present today is a paper that's in review. If the preprint is available, it's taking forever to get reviews back. The summary of the result is that we're looking at allele-specific expression, which Andy Clark introduced yesterday. And we're looking to see whether we can detect it by sequence alignment in hybrids. So you could take 2 species of Arabidopsis, hybridize them, sequence the seed, and try to determine is either parent overexpressed. And surprisingly, we can outdo the aligners with machine learning. I was very surprised to see this result and rushed out a publication. I've seen many papers where the machine learning results are overly optimistic. So I'm skeptical. I hope you're skeptical. If you'd like to talk to me about it afterwards, I'll welcome your insights. But I've tried hard to make sure this result stands up. So a little bit of background. My collaborators work on Arabidopsis seeds. It's well known that there's epigenetic phenomena going on in the endosperm. The endosperm is the food for the seed, and when you have a hybrid, the flower, the mother, would like to shut down this seed and not waste resources on it. And many seeds come out tiny from hybrids. But the pollen grain has a genome that has something to say here, and it tries to downregulate maternal genes and keep itself well fed. This is a well-known phenomenon. There's many papers, and we have a few papers about this with collaborators in Norway. So here's the protocol for this experiment. On the left is the training, on the right is the testing. So we take RNA-seq read pairs from Illumina. We're using public data for this paper, and we align them to 2 references. Let's say you have horse and donkey, and you were preparing to analyze a mule. So you take every read pair, align it to the 2 parents, capture the 2 alignments, extract features. We extract 53 features per alignment, feed it into some kind of classifier, and train that classifier to choose the right parent. That's the training mode. In testing mode, we do the exact same thing. We would take hybrid pairs, align them to 2 references, and let the classifier choose. That's how the experimentalist would use this software, but in fact, we can't measure the accuracy that way because nobody knows the origin of the hybrid reads. So we, in fact, make a simulated hybrid. We mix together the horse and donkey reads, keep track of which is which, and see how the classifier does. So we test on different reads than we trained on. So we're taking each read pair, an Illumina read pair, aligning it to both references. We get 4 subalignments here, and we've done this with salmon, bowtie, HiSat, and star. We extract features, and our features include things like mismatches, indels. They exclude things that could give away the sequencing run. So we exclude anything that could give away the read length, the insert length, because individual sequencing runs tend to have characteristic lengths after trimming. So we're trying, and if we include those features, the model does even better. So we're excluding those features on purpose. And as you saw in these results, these are 3 species that we tried, 3 hybrids that we tried. We're using public data, the public references. The aligner, note the y-axis starts at 50%, which is random guessing. The aligner is not doing so well, which was quite a surprise, but looking at why that is, the 2 references in every case are of different completeness and different quality, so there's map bias. And also, in many cases, the 2 alignments are equally bad, and the aligner just makes a choice. We can improve on the aligner, as shown in orange, by examining the alignment scores output by the aligners. So that tells you there's some room here to improve. And then finally, if we give all of the features that we extract to the machine learning model, we can get up to 90% accuracy. I want to stress that this is for a very special case. We're not doing better than bowtie and HiSat and star in the general case. We're in the very special case where you have a hybrid and where we can train the model on that specific hybrid, we can outdo the aligner. As a final test, we took a different horse and a different donkey from the same lab, but we're testing whether the model generalizes. So it's been trained and tested on 1 pair of animals. We then tested without training on another pair of animals and got similar numbers. So we think this approach would work in a lab that's studying hybrids. And here's just a few more details. Which machine learning models did we use? All of these performed about equally. So we used random forest because it could tell us the features that it relied on, so it was the most interpretable. I think we could do better with more work if we reached into the aligner and pulled out more features, but we're just relying on the features that are output by the aligners. We found 53 of them. We get features from each sub-alignment of each read to each parent, and then more overall features. We use high quality information from the read itself. So the sequencing read has a quality score per base pair. So we differentiate between an indel at just anywhere versus an indel that has high quality bases around it. And those features came out to be important in some cases. This is the advantage of using random forest. It will rank the features, and on the right you can see different features were selected per hybrid. So the model is really specializing on a hybrid pair. There's some redundancy. For instance, parent occurs in each list, and parent is the aligner's choice. So the model is taking into account what the aligner thought was the best alignment, but using other features to improve it in some cases. These are the species that we used. We used all public data and public references. In every case, we made sure the Illumina runs were compatible. And these are the aligners that we tested. Thank you very much.
We do have time for 1 or 2 questions, if anybody has any questions.
So,
Sam?
did you do anything with alternative isoforms?
You just repeat your question, just.
Did we look at isoforms? So we have the transcriptomes. It should include all the isoforms. So no, I didn't look specifically. What should I be doing?
I'm just sort of wondering whether there could be different transcript use of the two species, so that might be one of the scenarios too.
Ah, thank you. Okay.
Yeah, thank you so much. But we do not have Govind. So, do you want to introduce him, Soren? I can do it, but I will be woefully understated.
Let's bring him.
I did email him. So we are going to have to cut the refreshment break, so if you need refreshments, now would be a good time. Because I have,
Yeah, we're doing Misha, right?
Yep.
You can from the podium.
so do you want this?
Yeah. I'm fine, I have it.
Okay. I don't need to introduce you.
Are we ready? All right. I am delighted to introduce Professor Misha Gromov from the Institut des Hautes Études Scientifiques in Paris and also from New York University. So Gromov is without question one of the most inspiring and famous mathematicians living today. He is the winner of the Abel Prize, which is considered the Nobel Prize for mathematics, or one of them. He has a long list of awards, including Oswald Wellman Prize in Geometry, Wolf Prize, Boltzmann Prize, Kyoto Prize, Nemers Prize in Mathematics, Boyer Prize, and the Abel Prize, of course. So the list of topics that he made seminal contributions in Wikipedia is several pages long. In particular, he worked on Ricci flows and hyperbolic geometry and a number of very fundamental areas of mathematics that he contributed with seminal results. In particular, Riemann geometry, foundations, and hyperbolic geometry I would highlight. It is Gromov's Hausdorff measure is something used in computational biology group here at Brown as an important method for deep learning in genomics. So all over he made tremendous contributions, and we are very excited to hear Professor Gromov's von Neumann lecture today. Professor Gromov, you're next.
Thank you very much. I hope you hear me. Yeah? So, and it's my honor to speak on these lectures, because von Neumann was a great mathematician, and besides being a great mathematician, he was one of the few of us who could relate mathematics to something else. And so I'll do such attempt in my lectures. And then before I show my slides, I want to say that this is not mathematics, it's just what I want to say. It is what precedes mathematics. It's attempt to make, imagine mathematical model of human mind or some aspects of human mind, and really kind of imagine what it is mathematically. And I don't think it's the final answer, but this kind of direction I think is interesting. And so let me now to show my slides, and let me see if I can do it. To share screen. Somehow I have to find the button, but somehow it disappeared. Here it is. Now you see my screen, yeah?
Yeah.
It's okay?
Yeah, we can. If you
Okay,
can.
good. So my point is that what happens, fundamental processes in your mind are mathematical, and they are invisible to you. You are not supposed to see them. And there is a good reason why you are not supposed to see them. They're blocked from your conscious mind, roughly for the same reason why mitochondria kind of separated, isolated from the rest of the cell, because its products may be poisonous for the rest of your mind. Something happened there. You are not supposed to see that, but there are instances when it comes to the surface. And I start with this example, which I think absolutely fantastic. This is not example. That's just a picture how you think your mathematics and your mind interacting. Yeah. But this reflection will be part. Some moment I explain what reflection is happening there. And here is two formulas. And this is some phenomenon which happens in mathematics and was very particular. It was Ramanujan who read some book, and then what he produced. And so, and this transformation of symbols he received from the book, a symbol which he wrote, I claim, suspect, I know how to say it, it is a mathematical phenomenon. It cannot be explained by by words in any simple terms, right? It's just, and if you don't understand this, and refuse to understand it, then you are infinitely far from understanding how our mind works, in particular how it interacts with language and with mathematics. I emphasize 3 points of understanding. These are mathematical, language, and science, and this is twofold interaction. This is why I put these mirrors, and these are part of the interaction which characteristic of human beings is not presented even in higher animals, right? Only in rudimentary form, of course, there is something you can speak about language and mathematics, but not this kind of mathematics. And usual, sometimes objection which can be made. You see this kind of, looking at them immediately see what understanding signifies. How much you want to understand these formulas, depending on qualification and knowledge. So personally, I reasonably well understand the first formula, but the second one, I can kind of understand it, but of course I don't understand the deep meaning of that, which only available to people who knows it well. And this may be an objection, saying that this is a kind of isolated phenomenon, it's a rare phenomenon, whatever. But here I want to say that this is exactly wrong application of probability and statistics to this subject matter. That much of what I say justified by the fact, but statistical approach common in physics and borrowed to other sciences based on the following principle. And this mathematical principle, that average and typical is essentially the same. And because average, what physicists and everybody else can compute. But this you cannot see, yeah? Experimentally, you see kind of typical behavior. And this is kind of the law of large numbers, the simplest manifestation. And for the moment, there is large body of mathematics analyzing it. It's called concentration phenomenon, which was emphasized, first brought to the life in this form by Paul Levy 100 years ago, approximately, and then was popularized by various people and brought to the focus. And this remarkable phenomenon, which is non-applicable to biology or to psychology. So you cannot think you cannot exclude probability, but you have to change it radically and forget about this principle, or rather not to forget it, but know where to use it. Of course, it still has without it, very hard to use probability, but it must be used in a very different way. And so this is exactly because it is exceptional phenomenon. it is should be understood more than average phenomenon. Another example, if you look at the sky and see the stars, if you want to understand them, you have to first understand supernova. And these are very rare and very exceptional from our perspective, though of course they're very common in the universe because there are many stars. Because, as you know, no matter how rare they are, our life depends on them, not first of all, more than maybe than on such stars as our sun. Yeah? So, in the same way, and this basic process of understanding seen in this working of Ramanujan and his brain more than on the average. And at least I don't want to explain exactly what this. I can make the hypothesis why so, but it's another issue. But so this is what we have. What kind of mathematics you expect? You don't expect to find explanation of your kind of mental process in understanding, and we rather kind of, but rather just to find a framework where we can talk about them mathematically and kind of objectively. And just the two inspiring example, of course, is this Galilean symmetry, which define Newtonian mechanics and most what we see mechanically in this world. And I emphasize non-commutative. So it must be understood. It's extremely difficult to grasp intuitively what is the structure of the group of SO3 or of rotation of the space. It's non-commutative group. And if you look at the best mind of the 20 years ago, like earlier, they have absolutely kind of, on one hand, practically applicable, but no conceptual understanding what the group is, because existence of such a group, compact, non-commutative group, absolutely beyond our comprehension, no matter what you say. It's just if you take a physicist's point of view, you just describe these properties, it's impossible. And this, by the way, true about much of the mathematics. Yeah, mathematics on any kind of significant level is science about impossible phenomena. On the other hand, another example is opposite, where we have something very simple. Mathematically, it's just exponential function, and this mathematical part was understood very much by Darwin. And it's amazingly, it was not properly grasped by mathematicians at the time, but Darwin, on one hand, he didn't know multiplication table. He made mistakes in his computation. On one hand, on the other hand, he had mathematical intuition. And his intuition, and probably the same as Wallace, but I haven't read enough Wallace to say it, he understood that this is the right framework to describe evolution. Again, contrary to what some biologists say, it doesn't explain anything in evolution, but it tells you how to speak, no more than just the fact that law of motion obeys differential equation, but itself explains nothing, because you have to know the equation, you have to solve it. And even less so, this is natural selection plus explains evolution, but it allows you to talk about this in a constructive way, right? You know what are parameters, what to measure, what to look for, etc. And the point is, mathematically, you have exponential growth, you cut it off. And this is mathematics you can study and was pursued, of course, quite significantly. Now we turn to our mind, and so just what we have to reject is this terminology. So we have lots of self-gratifying words about how smart we are, and all these words, and I think they, exactly because there is a good reason why we say them, because so pleasant for us and self-flattering to say them, they must be absolutely irrelevant. I wouldn't say they're wrong, but certainly irrelevant to our understanding of our mental processes. This was confusion created a lot. There are more of these words, I don't want to repeat them, but all these words about how creative, rational intelligence we are, and it's some of them just wrong, some of them just meaningless. And the dictionary, what we want to use, what I said, is universal, some universal entities where we can start doing mathematics. With creativity or intelligence, they have no mathematical counterparts. But all these words have many mathematical expressions, and we want to look at them. And so let's look at examples. Yeah, there is some, I said, there is a constant probability. And which in principle can take into account some correlation due to the inherent symmetry in biological system and in mental system, but symmetry very different from the one which we have in physics, of course, yeah. And but this is beyond the scope of my talk. But here is two examples, and this is the one example. You have the bug on the leaf, and so what you have probably observed as a child, just that if you look at the bug on the leaf, it spends much more time on the edges of this leaf, yeah, somewhere here, and even more so, but not proportionally much more than inside. Really, just even in absolute time, not to say divided by area. So why it happens, right? And you say, I want to say, this displays kind of one of these basic principles of how brain of this animal works, and our brain works very similar. I haven't have these pictures here, but if you look at how your eye would do this, if you look at this leaf, your eye will behave more or less in the same way. You spend more time at a particular point here, at point here, on the boundary, but less than inside. No, but your eye will be spending some time along this line also. And so what is mathematically different? And there is one obvious things, and this is one of the universal you have to use, that amount of information you receive here is different. Here is the boundary. So here is one, here is another. So we have being present here, receive one bit of information. When you hear, if you forget about this particular pattern, so there is no information. So empty space is just zero kind of information coming to you when you move. And here you can kind of being on the boundary, it's more interesting. I interpret it in this way because it's more informative and more interesting. Of course, information I use in a formal way, right? It's a mixture. Of course, I'm saying the word bit, but of course, it's information relative to the system which observes it. So it's not information of a Shannon type. It's a questionable entity which is unclear if properly definable, but in concrete situation, you can observe it. So this is the one instance, right, of a mathematical pattern which is present in your mind. And so it is an understanding of edges of information. And so and it's kind of mathematically the whole mathematical theory you can imagine when you perceive world, perceive geometry in terms of so-called information you receive. This, of course, needs formalization. Another example, probably many of us familiar, is that how, by again, very formal process, ants as a community behaves in an intelligent way. So again, interesting feature mathematically here, that all these ants are, for this purpose, identical. So they behave in the same way, right? And so, and this defines this particular nice behavior, namely when there is a source of food, and some ant find the source of food, they come there, or several ants, and eventually they find shortest path to that, and everybody goes along the shortest path. And how they find the short path? And this is interestingly because it answers, the way they do it, answer to the famous kind of quotation by the Haldane. So Haldane was one of the smartest people ever working in biology, or in general, one of the smartest scientists of the last century. And what he says, yeah, he didn't like the idea that something was, his brain was, his mentality was governed by chemistry in his brain. He definitely resented this idea in what he says. But exactly chemical smell define behavior of ants, because what happens when they go to somewhere, they trace they leave some particular smell along their path, and so if the path is shortest, given equal condition, there will be more ants going there, so intensity become maximal. So the shortest path has maximal intensity of smell, and therefore being preferred by following ants. So it's extremely simple algorithm, and again, conjecturally, something like that be happening in your brain. Now, I must say, I'm not so much concerned what's happening in the brain and how this mental process is being implemented by the brain. There is a big gap, right, between the two, and this is not my business to fill it in. But the question is, what is in between neuronal activity, which we kind of understand, and our mental behavior, which also we observe. But the point, just I am kind of underlying what I'm saying, there is intermediate stage, which is neither of the two, which is not expressible in the common language. It's not something like your dream or your subconscious, nothing like that. It's really very far from any conscious perception or any kind of perception. And it is like behavior of this ends, yeah. Right, so we are not interested exactly how they produce these pheromones, which you put whatever, but the fact they are there, and then there is very simple trick. Another example again, which I find especially nice, is that a baby animal pretty fast learn how to distinguish goose from hawk. If there is another goose flying over them, they don't pay any attention to that. But if it's hawk, they immediately run into cover. How do they learn that? And you know, in artificial intelligence, there's a lot of talk about the top down, down top kind of logic. It's unclear how have to classify that. Yeah, but the point is, it is the exercise maximally abstract reasoning. You see, it's not concrete, and then they don't generalize. It's neither. Yeah, but so how they do that? It was actually, I think, it was Tinbergen and other people were involved in Tinbergen. He formulated the right conjecture, and it was confirmed. And they don't have in their mind picture of a goose or hawk a priori. But what they are able to distinguish goose from hawk. So they know the shapes are different, right? So it's one thing. They don't know what these are. They know they may be similar or different. And again, it is a very abstract general principle. It's number one. Secondly, what they know, some event is frequent or rare. So they have different events, something flying over their heads. They see something. And they know some of these events similar and some different. Right? So you don't have to know what events are. What you only have to know some are similar, some different, and some repeat themselves. So they can see repetition, and then they can count. They don't count in numbers, but they know event frequent or rare. And then it's very easy strategy. First, they always run into cover. If something happens, something changes, this is another fundamental phenomenon of the life. Mostly nothing changes, right? Things changes are rare. Boundaries take smaller amount of time and space than interiors. This is true in our geometry, and it's true in our life. And there's a kind of famous story about Fermi, who was making bets during the Second World War, always winning. And his statement was, nothing's going to change. And so usually you win. But on the other hand, you know, if you always follow this strategy, eventually you lose horribly, right? So, but locally you win. And so the same about, so things eventually change, yeah. So it's very dangerous to think they will never change. They do change. It has to change, but most of the time they don't. And so they run always, because most of the time nothing flies over their heads. And then if something happening too often, and there is a probably built-in idea in their brain, what does it mean to sufficiently often to ignore this, then they don't run away, because hawks appear rarely, right? Because there are much fewer hawks than geese, because hawks have to eat geese, and so there are 100 geese at least for each hawk. So that's and this is actually how we function. Our brain works the same way. So again, the point that I want to make: when you can use mathematics, you have a hope to use mathematics, because there are some universality in the processes which determine your mental states and mental behavior, whatever they mean. And so you turn to that. Now I did it only because I couldn't put my to put pictures in the right order. But these, by the way, nice pictures, and you see, they're kind of pleasant to look at for some reason. And it's another story why. So also mathematical, by the way, regardless of their shape. And then the last one is some discovery made by Douglas Spalding long time ago and usually ascribed to, but to different people, unjustifiably. And this is a imprinting, that how baby chick knows who his or her mother is. And this is abstract thinking. Again, it's the first moving object. So again, this concept of the first and moving an object. So there are three concepts, very abstract, of the same type we had before. First moves object. Something changes its state, constant or not. And experimentally, and it was much popularized by Lawrence, who made these experiments, and you can use any object. And this, by the way, was known before Spalding. It was observed by people before Spalding, but he was the first to make experiments on the ladders. And he's not as commonly known as he deserves. He was contemporary of Darwin, and he was really a brilliant, absolutely brilliant mind, and he died young because he was poor, and usually, like most people of the time, died of tuberculosis. And I think he kind of deserves to be remembered by psychologists. Psychologists, by the way, when I spoke to some of them, they hate his name because it's not kind of science they like, yeah. And you see, it's very mathematical, yeah, by the way, behavior of these animals. It's another, by the way, how it's happened, they go in one line, how they know Euclidean geometry. It's another point of it, because the laws of mathematics so simple. Now come the following quotation by, there are many quotations by Einstein, and my impression and actually this also applied to many quotations of Darwin. Most popular of these quotations are just fake. Yeah. For example, this one is, for some reason I want to bring it up. It's something to do. It was what I'm going to say, but certainly, obviously, it's not Einstein. So if you have very common thing ascribed to a great man like Einstein or Darwin, it will be again for interesting psychological reason why such sentences being ascribed to great people. But what is the difference, however, between knowledge and understanding? Because understanding, because we speak about understanding, comes in many flavors. And any of us are probably familiar with that you're at a lecture. Yes, for example, if it will be mathematics, physics, bioinformatics, and if you really understand what happens, you're tired after 1 hour already. But if you listen something like anthropology, you're not getting tired. You can listen 5 hours in a row. And the reason is that it's combinatorially different things which happens apparently in your brain. I don't think it was ever checked by maybe visible in MRI, because the amount of blood definitely different. You probably feel exactly how much energy your brain consumes, depending on what you do. Because when you learn something, you create much more connection in your brain. It's a graph, kind of first approximation. It's a network, and if it is just it's just knowledge, you just have kind of a tree, and then in this tree if already you have ready places to put these things, you put it there. If there is no ready place, of course, you get hard work. But even a subject you know very well, like for me, it would be mathematics, it's the hardest to listen because it requires maximal amount of transformation in your brain. And now, so what we learn and what we come out of that. And so the point is, so what you have, what comes to your brain is something like that, and nothing else. So you can forget, but of course you cannot forget it, otherwise it's impossible to describe it. But temporarily, we forget the subject matter of what we want to learn. It may be mathematics, language, or science. I will be emphasizing mostly mathematics and language, which is just before our eyes. And so you just have parallel signals going into you. So it's essential, there's several parallel, but of course it's difficult, and sometimes we will pretending there's only one sequence of symbol going into your mind, to your brain. And that's all you have. So specificity of the world disappears, and you have to recover this. So the point is how to looking at that, recover the picture. For example, it may be something coming from your eye. Of course, it's not a once flow, it's several parallel signals going simultaneously. Languages better. Languages have very simple one-dimensional flow. The amount of your brain power involved is minimal. And this, by the way, specifically human, especially when it comes to understanding formulas like that. Yeah, you cannot claim, for all my respect for various animals, there is no animals who understand it, even among us. For example, the second formula is understood maybe by 100 mathematicians in the world who understand the second formula. The first formula understood by most mathematicians, and the second formula. However, I'm saying it is definitely very specific. So why, and I'm saying this might be much simpler than understanding something else in our mental process, because, for example, this behavior, this kind of this has longer history, right? Evolution of these basic reflexes was working probably at least 100 million years. And on a rather large population of animals, especially when it's coming to insects, yeah, it was really lots of insects. So it going on quintillions experiments that nature could make. However, if you look at us, at human, that by the most optimistic estimates, when we were separated from chimps, and it was about 5 million years ago, the number of tries evolution had was well below 1 trillion, right? Because population, for the most part, was just several thousands. There was some bottleneck when it was just 1,000 roughly. It was never above 1 million. And so the number of children born, say, there is one, in this population, one child, each parent have one child every year. So the number of births and deaths was tremendously small. So, and however, with this very small number of tries, some very sophisticated programs, well, sophisticated as we perceive them, and claim they're not sophisticated, but they look sophisticated. Programs were produced which allowed us to make this, say, understand language, understand mathematics or science, which is not characteristic of animals, and which even for human requires a particular effort. It's not natural for us to understand mathematics on high level. It's counting 1, 2, 3 up to 5, okay, but that no more than that. Then you have to do something in your mind, but your mind has this universality to do that. So it must be very, very simple programs. So by random choice, you make, 1 trillion tries. With trillion might look big, with tiny number compared to number of configurations and possible programs in very few symbols. And this is, it makes you hopeful you can do it yourself and guess what it is. And so what I will be describing is a skeletal of what you can call understanding, yeah. And from the skeletal, I'll give you only a rough idea of the creature. This is actually a skeleton of a whale, and certainly hardly you can see a whale, but, and so what it is. So what are basic elements out of which this understanding is built? This is kind of in the first approximation of that. So what I want to describe at this level is the next would come from a physicist's analysis of a language. So there is, this famous manuscript, Voynich manuscript. It's a book. It's about 300 pages, written in unknown language, unknown topics, and nobody can say even if it's human language or it's a hoax, right? And physicists analyzed the statistical distribution of patterns and found out, well, it agrees with human languages. And so this is the first what the physicists do. They see repetitiveness of certain strings and their correlations, and well, there is some pattern. And we have to imagine the next level of that, which tells you more about the structure of the language. In particular, if you can guess if such sequence of symbols carries some meaning or it doesn't. So, but of course, meaning is again an interesting word, which was much discussed by linguists, and so we will be careful. So these words will be using in two ways, and sometimes usually. By the way, I'm not certain when I started how long time I will have. I think I have 55 minutes, and I think I spoke already 40, right?
Can you hear me,
Yeah.
Dr. Verma?
Yes, I hear you.
You have, if you could finish around 10:40.
10:40, yes, this is what I expected, yes.
Perfect.
Okay, thank you. And so out of which we make our network. So it will be network with extra structure. It will be hierarchical multi-scale colored network, right? So it's network, then network of networks, network of networks, and also there will be colors. So networks is binary relation, and colors are unary relations. And some of them come from within, like what we see on this animal. There is no network. We see this atom of thinking. This will be there, but some of these nodes of this network coming from outside, like we see, like words. Yeah, this will be one of the units. So, and this is a basic conjecture which can be objected. We take discrete model, and apparently there is a good reason for languages. Language is discretization of our thinking, and all we know about our conscious thinking is by language. Everything else is just you cannot say, so it's there or not there, we forget about that. So it is discrete. So it will be something discrete. However, many of these units may be a little bit vague, like it happens with genes. What is a gene? It's a little bit vague notion. Which particular segment of, even when we say it's a segment of DNA, it's already kind of different from a gene of classical genetics, yeah, which corresponds to some protein or to some visible feature, right? Even there, which segment you regard as gene is vague, yeah, and it's okay. So it will be discrete structure, but with certain allowance to be stochastic or ambiguous in some way. So these are basic units. There are units, and then there are relations between these units. And there are two types, very different kind of relations, fundamentally different, though both not quite binary, but at this stage, think about them as binary. One of them, similarity between certain units, and we don't know a priori what kind of similarity, right? But just something similarity. In a second, they say what makes them different. And co-functionality. And the fundamental difference is that similarity doesn't depend where they appear in our perception. They may be very fine, having nothing in common, but still have words in different books. It's still regarded as the same word or something. For us, it's just something similar. But things which are co-functional must be in part of a bigger unit where we work together. They cannot be separated much. So there are the concept of two fundamental different concepts. Of course, there is another delicate situation here. You see, I will be speaking about using mathematics, but I'll be very careful in that way, that it's not any kind of, I use much of the standard mathematical language, on the other hand, none of any specific mathematical idea or theory, which is sometimes you can see people take their beloved part of mathematics and try to use it for modeling, say, our mind. And I think it's a priori wrong idea, because, well, we don't know what it is. And most likely it's unknown mathematics to us. It's particularly concerning probability, I would indicate what are obvious modifications we have to make to probability will become not so elegant as we have, and to apply it to languages. And you know that Chomsky was always objected to using probability in languages. On the other hand, today the way to make artificial system of translation depends on statistics. It works very well. But the point is what kind of statistics and how it's being used, right? So you might be careful about that. So, and then just one particular point is that one structure we must be very careful with is order, because the way I understand it, if you say A greater than B, and then B greater than C, then A greater than C, this cannot be explained, formulated in a certain mathematical language, right? For the reason, because you cannot order two entities, define their order, unless they already be ordered, either by being written on the line, on the blackboard or paper, which has internal geometric structure, or in time. And the time is your internal psychological time. So you assume that your time and time of to whom you speak, they're the same, and we know it's tricky because we are not aware of true time which happens in our psychology. Therefore, order and again, experience of most of us shows that order comes later. We always have mistakes. Who is sign? You see, if you say A, B, the order sign between them, how you know how it's directed? because you have to say beforehand that B goes after A. Unless you know that, you cannot say what sign of order means, right? So this is a very essential point, and I think it's not properly appreciated, which actually underlies the more subtle logic in mathematics, like Gödel theorem, crucially depends on that, on this argument. You bring in this artificial order. You cannot formulate, or rather, you cannot make the main step in the Gödel argument unless you have that. And so, which, in my view, make it not that rigorous as people usually say, because it depends on, it's more or less of the nature, the same as when you say some people knows left or right action of the group on the space. You know left from right. You know your left hand and your right hand, but we know that you only can say it because there is break of symmetry, violation of symmetry in the weak interaction. And if you don't know that, you cannot tell left from right. And so your language depends on this physical phenomenon which was discovered recently. And so it's a little bit absurd to take it seriously. Yeah. And indeed, it is different in your, so there are different levels of mathematical sophistication inside of this network. And the order is much higher level than just similarity or co-functionality, which is symmetric when you have a non-symmetric entities. So again, so you take mathematics very, very carefully. You have to be more critical to logic than logicians are on one hand. On the other hand, this mathematics must be not logical mathematics, it must be really mathematical phenomena which must enter the game. So my point is here that there is in common, what join together our mentality, our brain, our mind. Brain, I don't know what brain is, just your mind. What you can do, you can prove this theorem, it's a remarkable thing, and mathematics itself. And they both about impossibilities. What happens in mathematics for any rational mind who yet understand mathematics but doesn't know it, it would be impossible. Any non-trivial theorem in mathematics is something about impossible. You can prove from general principle, it's just probability of this is close to zero. And that's exactly what makes mathematics interesting. And the same about our brain. If you could say to somebody educated entirely in physics that there is such things like living entities or thinking entities, it's absurd. So exactly all physics based on hidden, unspoken assumption, that's impossible, right? And he's very, I dislike when you bring this up. And so it is about impossibilities. So with mathematics, you must be careful. You don't use mathematics, you only use mathematical way of thinking. But unfortunately, we cannot do it without using what we have. So, and so our learning process, so not our conceptual understanding is inseparable of conceptual learning, because this network is a dynamic entity, and we don't know what it is. And here is another basic mathematical idea we borrow from mathematics, that we don't know how to say what particularly this is. What is this network we want to describe? And it may be very, very much from person to person, from situation to situation. But the class of this, universe of this, must be, in my view, describable. So, and for example, it's very difficult to say about some physical constant what exactly value of this constant is. But you know, it's a member of a community we understand. It's just a number, and we know what numbers are, assuming we take mathematics the way it is. Yeah. And here is an example from mathematics. Just I want to, because this is a not kind of, again, not properly known to mathematicians away from this field. So he sees here, this is algebraic curves, the Radis's formulas. And then people, mathematicians, worked and wanted to give you different, what are the curve? What is this algebraic curve? And this was kind of foundational algebraic geometry. And there was a book written by André Weil, where he summarized kind of principle of mathematics developed up to 1950. And so in 1956 or maybe 1960, he wrote his book, Foundational Algebraic Geometry. And then, maybe 2 years later, came the Grothendieck definition, what's algebraic curve. And Grothendieck definition, and the definition of André Weil was very natural definition It was a set with certain properties, blows the definition. And I remember when I was learning it, it was so nice. Just you felt almost yourself, you could make the definition. But then came definition of Grothendieck, and everybody, of course, at least for beginners like myself, was shocking, rather absurd definition, because he was not defining a curve, but defining some category of a fine scheme, But then time comes, and we know what everything natural, nice definition by Andrew was absolutely inappropriate. It was just historically motivated. But a Grothendieck, now everybody uses only the Grothendieck's point, and it's one of the essential ideas. You don't define so much this curve. You define community of objects, a very large community. And this community of schemes or some category of scheme, and only there you can locate what are these algebraic curves, because you cannot define them in isolation. And therefore, it's much easier. And to say what a particular curve, what this equation, it's come much later stage. And you can think, aha, this is very abstract, it's very difficult. But this is, again, it's not, I don't think it's correct. Say, thinking of category theory of mathematics is most elementary thinking. Children think in those terms. Physicists, classical, if you translate what Boltzmann, how he was describing ensembles of particles, exactly was thinking in these terms, and was criticized by von Rieger, by mathematicians, up to a certain point, because exactly they didn't have in their position this kind of things. As Grothendieck emphasized, zero, not very difficult thing. It takes century the people to come to the idea of zero. And this is the same about category theory. It's the same level of abstraction as zero. It's on the bottom of our mind, but it doesn't come to the open. And I think there are other things in the bottom of our mind, some logical thing happening there, which don't come to the open, and they define what I want to describe. And now, so what is understanding? As I said, understanding is particularly language. And language, of course, very nice, because you have huge amount of language now on the internet, and you see how much you can understand it, and what this understanding means, and what language is. And just this instance of that, so this definition was algebraic curve was a big success in mathematics. Mathematicians were able to define what is algebraic curve. But certainly mathematicians failed to give definition of a language. And there are some mathematicians try to say language is a set of strings, or language probability measure on the set of strings. And this, of course, is linguists would laugh at that justifiably. Yeah. And so what is the language? And so I say, well, this is a kind of symbolic representation of reality or some other kind of words. So this is a good example, yeah. So you understand this sentence, but what the hell means you understand it? Right? This is exactly in what way we understand language. And this is kind of quite bona fide sentence, a bona fide idea, but it's very tricky what you understand with an language. And I haven't brought enough examples, but I'll bring in a second more of them, yeah. And language is actually, transformation to language is very much how we understand even visual patterns. And visual patterns, it's another matter how we understand visual patterns. And there are two fundamentally different types of what we perceive and analyze visually. So I have only 3 minutes, yeah. And in our vision is very much determined by a life system, which is completely different from one which is pure nature. Here is an example. Yeah, so you look at that. So from certain formal point of view, both pictures are very much the same. It's the same colors in the same shape. But they are fundamentally, the question is, are they fundamentally different? Or is just because we know the face is the face, right? So can you tell the difference? And my guess from individual, from these two, it's not enough. But if you have 10 like that, Martian is not a Martian, but kind of what you see on the moon and the Mars is like that, and or not necessarily faces, but faces in animals, and then you can see that there is fundamental difference between them. And so your understanding applies only to this thing like faces, but doesn't apply to this kind of landscape when there's a random landscape. So the way you can understand something depends on what you understand. And something will be beyond your understanding, and some mathematical method like Fourier analysis, wavelets, whatever, can tell you much more than your mind. So now I wanted another idea we can borrow from mathematics, only say how mathematics may be used. That there is certain commutativity. This is a kind of fundamental concepts in mathematics, and particularly in operator theory, which describe physical systems, is commutativity. And then the point is that most or many rules of grammar can be learned independently one from another. The order doesn't matter. And this is another point. The order is kind of a tricky point. And therefore, our time, and also it's partly due to the fact that your brain works in parallel. And in parallel means it has different structure for its description. You see, sometimes say, oh, your brain is parallel, give you extra power. We know computationally it's no. Sequential computation is more difficult to make parallel than the other way around, right? So parallel in a way more primitive on one hand, and this is true about many things in your mind. It's more primitive than you do in computers, but on the other hand, it gives you different possibilities. When you have this two-dimensional picture of your time, you can make pictures there, which you cannot do so easily on one-dimensional, and that is the point, yeah. So you bring this artificial structure. It's there in your hardware. It's parallelism. And over this, you can introduce something else. Yeah. Well, so my time is running out. And so I show you just some example of sentences just to amuse you. Yeah. This, by the way, true quotation by Einstein, yeah. And this interesting point, and some people say that means that there is some great design in the nature, but it's because one of the things why the world is comprehensible, and this is crucial for our analysis, what you do, because mostly it's empty space. And so nothing happens except in the boundaries. So there are some interesting feature of the signals which we receive. Again, very simple. Yeah, for example, this one. So this is a famous story. And again, I just want to emphasize a mathematical point where apparently there is no mathematics. Right, so you read it, I hope, and so down to Ansel, we solve. Right, so again, this is the similarity, the same man appear in different contexts, and this gives you a link between different parts. So this similarity may link together. This again, the similarity linking different events is characteristic both of what happened in biology and in human minds. So, now it is something about meaning, but I wanted to show you for the last some simple examples. Yeah, it's again, yeah. How this relate to this. So all I describe here, there are some mathematics. One algorithm, maybe I want to say, which you are well familiar with, and which is fundamental in this, is B clustering. Of course, in bioinformatics, you systematically use B clustering, and it works remarkably. And this also works in, I believe it's one of the fundamental mechanisms which works in what we call understanding. So this, but I want to show you example indicating how tricky is the idea of understanding. Yeah, here it is. Yeah. Yeah, here is a sentence again, and you kind of understand it. But this, I wonder, if you slightly change the words, if any present computer would pass this kind of Turing test. If we spoke about goat and then we speak to the cow or something like that, and So we see we understand the sentence in a very tricky way. Just look at this, and you see how it's different from understanding the subject what is written in the rest. So imagine we had that, and then we ask this question of your computer and see if it's able to answer you. And I bet you will not, because the self-reflection of the language in mathematics is one of the ingredients. Because see, you know, language, even more than mathematics itself, has this kind of self-similarity structure. It has any small part of the language can incorporate the whole language. And in mathematics, it's essentially a feature of the Gödel theorem, which is Gödel theorem, or rather proof theorem, says that mathematics is fractal. That tiny piece of mathematics can reflect the whole mathematics and can be repeated infinitely many times. And this is a structural property of Gödel theorem. And this is what we remember in our model. Our model must have this property, which is very, very delicate property, which is very hard to formalize. And this is one of the difficulties which we hear here. Yeah, that's another famous joke. Yeah, and again, we understand it. It's interesting again the basis for our understanding of the sentences. And so, the problem is it is really kind of almost purely mathematical, which is I don't know how to solve it, but I think I can formulate it. But if you have this class of graph, I can describe what kind of graph they are, which would reflect some reality. And the point is that if you find that, if you make the structure, it cannot be wrong. And this probably you know with B-clustering algorithm. If your B-clustering works successfully, it couldn't be by accident. Probability of accident is virtually zero. So, but I wanted to show some examples just for. So I'm here describing what properties we know this mathematical object must have. You can, on the basis of your experiment or what you know, there are some number of properties. It's typical of your mind. You have to don't go too deeply into that. Even very superficial property like I described, if they're satisfied by this mathematical model, it will be fantastic. You can construct such model because a priori such model is impossible. But in mathematics, many structures are impossible. So you look for mathematical structure, which model your mind. And if you can formulate this property, you think it shouldn't exist. But because we believe mind exists, and this should exist. In mathematics, it's okay. In mathematics, often things which you think don't exist do exist, or almost always so. And just to entertain you, I just give examples of certain sentences when, see, as usual, yeah, it's impossible.
Yep.
So one thing we have to be careful about logic. Yeah, this is an example. Yeah, Socrates' motto. My time is over, right?
You have like 30 seconds left or so.
30 seconds, yeah. But I wanted to show you some sentence just to make you. See, I didn't clean enough this talk. I was. Well, probably of course, I didn't hope to come to the end, but in the end, I was showing something amusing. I lost it. I was just explaining. No, I couldn't find what I wanted to say. But yes, I want to give some extra examples when you see sentences of different kind and how you understand and completely, the understanding of one and other have nothing in common. And so just I want to emphasize a point that your understanding is not some unique entity. It's a member of huge community of mathematical objects, which we want to define. And these entities interact because your understanding is dynamic and exists in many stages and had this self-similarity similar to the one we observe, say, in mathematics and by Gödel's theorem. And well, of course, its description takes a long time, yeah, and it's conjectural anyway, because as I said, it's conjectural structure which potentially may imitate a model, what we call understanding. And this understanding, returning to my first page, we do know it is there. We either understand or don't understand it. And so there is difference between two states of the mind, understanding and non-understanding. And we want to understand the structure of this. So what is in your brain happens when you see and start understanding either of these two. Okay, we say. And of course, all what was, of course, in the brain of Ramanujan, but not on the neuronal level, but on the level of deeper logic, which generates such entities as these formulas. Okay, so I'm open to questions.
Thank you so much. We have time for maybe one or two quick questions. One longer question or two quick ones. Okay, I'll ask, I don't know if this is quick, but so Dr. Gromov, does your mathematical model of the mind have anything to say about philosophical concepts like consciousness or free will?
Okay, you see, at the very beginning, I said it, yeah. It's just I say these words, I have nothing against them, but they're not interesting. They just, you can speak about them forever. They exactly brings us in a completely different mode. I don't want to say anything for or against it, but they're not exactly a priori formalizable. The model which I have in mind is to understand and to improve the working of artificial intelligence, because we have these neural networks and they work fantastically well. But we don't know, we have no mathematical model what happens on the intermediate stages of this computation. And I think you can tremendously improve that if you know what you do, right? You don't do it in a blind way, and it's kind of blind search by iteration, but make it more organized. And what is this consciousness or free will? I don't know what it is. You can say, I actually personally, I don't know what people say about it. People say these words. For myself, I don't know what my free will or my consciousness. You have to be convinced, you have to learn that there is this concept and convince yourself it's there. I don't know, I don't have it. If you don't brought from the childhood, you say you have free will, you don't have free will. You have it, of course, you behave in some way, you don't have this idea. I think it's a cultural idea. These are both cultural ideas, and okay, like being good or being kind of free or whatever. These are okay, but I don't know. I just personally, internally, I don't know what means I myself, whatever. With deep philosophical meaning, I think philosophy with a certain mentality have this very strongly, and then pushes the idea on us. And I resent it in a way, because I don't want to be trained or manipulated by philosophers. Mathematics is, this formula is reality. You read this formula, it is a reality. And you know, as said Democritus, or at least, of course, he never said it, that there are only empty space, moving atoms, everything, the matter of opinion. And so this is a matter of opinion. And, sure. and it's somebody's opinion, it's of no interest to us. We only want to know the reality, which we can put your finger on mathematically or scientifically. They are very two different ways, but both, which, well, productive. And the other are not free will, yeah. Yes.
That's fair. Thank you so much. Let's have another round of applause for. Okay, next, we have another round of 3 more Zoom talks. So the next Zoom talk is going to be given by John Kleinberg. So John Kleinberg is the Tisch University Professor of Computer Science and Information Science at Cornell University. Whenever you're ready, John, please. There okay. You are.
Are you able to see? You able to see the screen?
You don't see your screen yet.
All right, let's do this, and then this. All right, can you see that?
Perfect. Yes, thank you.
Okay, great. So should I just start? Well, thanks very much. I hope you can hear me in the room. Thanks very much for the invitation to take part in this. Congratulations, Soren, and happy birthday. I guess I feel like I also obviously have to, acknowledge that seeing the upsetting news this morning and knowing that there were, I have friends in the audience who are even more directly affected and connected to this. It's a bit disorienting to just go ahead with the talk. But yeah, so I just wanted to acknowledge that, and my thoughts very much divided between these things. So again, it's great to be able to take part in this wonderful event. It was exciting even just to read through the program and see all the fascinating topics represented. What I wanted to talk about was some ways in which we've actually been using mechanisms, algorithms, ideas from computational biology to think about networks of online content, a topic that I tend to work on, have worked on for quite a while. And I think we all sort of know about the metaphors of internet memes or internet content, as having a kind of like, these biological metaphors that underpin the mechanisms for them. But maybe what's been less apparent is the fact that actually these metaphors you can actually make formal, you can make precise, you can make quantitative, and actually some of the algorithms that have been constructed on the computational biology side have actually proved extremely valuable as we've tried to map some of the content that we see on the internet. So I want to walk you through, and this will actually reach somewhat far back into the history of this work, to really walk through some of the ways in which these connections were actually made somewhat formal and precise. And it's a pleasure to be able to do this at this conference honoring Soren. I think I first encountered Soren's work in graduate school, and actually I can sort of point to the paper I think I originally encountered, which through graduate school really struck me as one of the wonderful examples of how you can take a very complicated phenomenon, and by building a very clean, a very elegant computational model and then analyzing it, building algorithms for it, you can really get insight into the situation. I first met Soren at the RECOM 1999 conference, where he was the program chair, and where a number of the other attendees were either invited speakers or other participants. So I learned through all of this time, I benefited enormously from my contact with these algorithms, with these models, and they informed my thinking about something that at the time I was working extremely hard on, which was the question of how information travels through the web, how information is organized on the web, how it travels through the web. Something which, again, on the surface, we know the metaphors between online information and maybe genetic information, but it was really striking to me how much you could actually make this connection precise, right? So when we think about web information, the web has always been balanced between these two metaphors, right? The web is a vast universal library of information, right? Pages with hyperlinks, and the web is as a giant crowd that reacts in real time to news and information, right? And this question of how does the crowd respond to information, how does it synthesize new information? These are very old questions, right? These go back to the early history of the theory of mass communication, to the early writings of Vannevar Bush on the origins of hypertext and hyperlinking and associative memory. These are very, very old questions, but it was only really in the last 20 years that we could actually try to measure how this kind of information spread at an incredibly fine-grained person by person level. And so that was something that I was very interested in from the beginning of my contact with these problems on the web. And around 2003, I was working with my former student, David Libben-Nowell, with whom I've had a really long and productive collaboration up to the present. And David had this very interesting idea for how we might, even in 2003, which remember was really before social media, even before there was Gmail as an email service offered, how we could actually try to trace the flow of information, looking from the outside, just looking from web crawls. And the idea was to take these things that were going around the internet at the time, chain letter petitions, and treat them as tracers through this sort of global social network of people sending each other email, right? So this is an example of one of the early chain letter petitions from the early days of the internet, where there's a header, there's an appeal, and then they're asking you to basically sign this petition if you agree with what people are talking about, right? So in principle, this is something which, is spreading through the internet, as we see many, many things now on, spread through the internet under all the social media mechanisms we have today, right? It starts at some person, it spreads, they send it to some people, they send it to some people, we all sort of know how things spread exponentially in this way. But how does this help us, right? So Dave and I were looking at this and saying, not only do we not have access to this, this is, the early days of the internet and no one has access to it, right? Google can't see it because it doesn't have Gmail. Facebook can't see it because it doesn't exist. How do we see it? And the insight that David had was hundreds of copies of this chain letter, when it was being mailed out to people, were also being posted to mailing lists that were archived on the internet. And because it was not only a chain letter, but a petition, you actually had lists of signatories. And so even from just simple web searches, you could construct thousands of these, right? You could collect thousands of these from the internet and begin piecing together, looking from the outside, the tree that it followed, right? And some of these activities might be recognizable to some of you in the audience from similar strategies in bioinformatics, right? So we find a copy of the chain letter. It has signatories A through H, and we put down a tree here. We say, okay, we find another copy. It agrees down to F, but then it splits, right? It goes to IJ. So, okay, it's a split in the tree. This one agrees down to D, but then it goes off to K instead of E. So not a split in the tree. We quickly discovered when you engage in this reconstruction activity that you have all the problems that we know from other places in computational biology where these kinds of questions arise. So for example, somebody goes into the copy that they're signing and they think it'd be funny to, change the second signatory to, the name of some celebrity or some political figure. And so you get a copy of the chain letter that just has some new name here that shouldn't really be there, right? So you have this kind of point mutation in this. Some people get the list and they think it looks too long. So they excise a big block from it, right? So they just take out B, C, and D, and you get like a jump edge this way. And as we went through this data, we discovered you had all these things and you had others. Some people thought the list looked too short and they actually copied a block of names and just duplicated it somewhere in the middle to make it look longer. Some people thought they looked too short. They had two copies, right? And they wanted to sort of sign something that was the combination of both. So they would actually take blocks and they would like merge them, right? They would actually do some kind of hybridization between the two lists. And so all of these required some kind of reconstruction algorithms where we actually were going back to, right, string reconstruction, assembling trees with errors. And when you do all that, we actually got this enormous tree. And this was really one of the largest cascade trees to actually be explicitly visualized at the time. This is over 20,000 distinct signatories assembled from 1,000 or more messages, right? And as you zoom in, it has this very striking structure, which despite the 6 degrees of separation that connect all of us, it goes very, very deep. Many things look like chains of, just single children nodes. And so there was a lot of mystery here to be figured out, right? It did not look the way we expected, right? Despite short paths in the social network, we have these very long paths in the tree. I should mention I'm in a hotel room where I'm going to be leaving in like a few minutes, but because I'm in the middle of traveling. So so we have over 90% of the nodes have only one child. But the funny thing is that this is actually the result of the sampling process in some sense, right? So one thing we could actually do is prove a theorem that says if I have a large tree, it has bounded degree at every node. And then with very low probability, some very small fraction of people, probably delta, decide to post their copy of the chain letter. And then that lights up the path back to the root. Then in fact, a 1 minus epsilon fraction of all nodes are going to appear to have only one child in the reconstructed tree. And this actually allowed us to do some other inversion problems where we could actually take the sample of the tree that we had here, and we could say this is just a snapshot, we could actually infer the total number of people who had actually signed this petition as the petition went forward, right? So you can engage in this inference, this reconstruction problem. Now, as we thought about these problems, we became curious about other questions, right? This was a single piece of information spreading, but we were really curious about the aggregate effect of all of this. And so again, if I'm sort of moving forward through history, thinking about the way some of these bioinformatics algorithms came to play a role in our work, we got to the 2008 US presidential election, and we really, for the first time, had a kind of data that allowed us to ask, can we actually map the news cycle of something like, like a US presidential election, using data from services like Spinner and other very early precursors of these large social media information repositories. Now, here we face a definitional problem, which is if the news cycle is the tempo of stories as they play out over time, what is a story, right? A story is not a topic that could run for a very long time. It's not an article that's much too short. It should be something that sort of lasts over the order of days, that it rises and falls and is replaced by some new thing in the news cycle. And so I was working with two former students, Yuri Leskovic and Lars Backstrom, Yuri and I worked out this idea that maybe a thing we could actually trace was phrases inside quotes, right? One thing that's common to almost all news stories is that they're constantly annotated with things people said in quotation marks. And so if we could latch onto a quote, we could watch how that quote traveled through the news, and in that way try to figure out something about the tempo of the news cycle. Again, we faced a problem because, again, every time you latch onto a string-like piece of information, you discover that it somehow mutates despite your attempts to control it. And quotes on the internet in news stories mutate a lot, right? So this is an example, again, 2008 US presidential election. And this was a paragraph from that Sarah Palin used over the course of roughly 2 weeks in fall 2008 as part of her stump speech on the campaign trail. She went around and she gave the speech many times, and she said it slightly differently each time. The news excerpted, different news articles excerpted different things each time. And sometimes also the news just quoted it slightly wrong. But she was also varying the language as she experimented with it. And so essentially this paragraph, right, this kind of ideal canonical paragraph that Sarah Palin was using on the campaign trail, and this is one of hundreds of examples we had, was essentially exploded into fragments over websites all over the internet, right? Each website had a slightly different fragment, and they didn't always line up exactly right. So in a process that, again, a number of you are intimately familiar with, but this time with quotes from the news, right? We had to go and we had to actually piece together these things to try to reassemble what was this actual thing that was being said so that we could then trace it and then try to map the tempo of this. And then at the end, we were able to present to organizations like the Pew Foundation, which monitors, sort of the practice of journalism in the U.S., this map of what the 2008 presidential news cycle looked like, right? Where each week there were certain quotes that dominated the news cycle and essentially gave us hooks, right? These things were tracers in the sense that any news article is this amorphous object that's very, very hard to get your hands on. But the quotes, despite their mutations, were much easier to sort of trace. And so we could follow this as it went through the news cycle. Now, in the process of looking at this, there's one final thing, right? So we've talked about transmission. We've talked about mutation. And of course, there's sort of a third biological analogy that sort of can't help but jump out here, which is the idea of relative fitness, right? Some of these quotes were really breaking through and were all over the news. And some quotes just sort of would fizzle as soon as they were being said, right? Why is it that some quotes are really get this kind of traction and other quotes just don't? Essentially, we're asking a question about a fitness function on the language that's being used in online news, in online media, in the way people share things on the internet. And so the question we had was, how do we try to estimate some notion of a fitness function on this kind of language? And so this was then in joint work with Christian Dănescu, Niculescu Mizel, Justin Chang, Lillian Lee. We went looking for sources of viral text where we could see some succeed and some fail and try to estimate something about fitness functions. And we looked at one more source of data, which I wanted to share with you here. This idea that movie quotes are, of course, a reliable source over many, many years of this kind of viral, highly memorable quotes, right? We have entire scripts, and most movie screenplays contain lots of dialogue that we don't remember, but certain lines we do remember. And so our strategy was to look at scenes in movies, right? Lines spoken by the same character in the same scene of the same movie, where one of these things became viral and the rest of it didn't. And to ask, like, can we find things that distinguish these, right? So just as an example, right? So this is a screenshot from the movie Star Wars, and many of you may recognize Obi-Wan Kenobi and a stormtrooper having a little conversation. Obi-Wan saying, you don't need to see his identification. These aren't the droids you're looking for. He can go about his business. Of these three things that Obi-Wan said, one of these was in our language much more fit than the other two. It ended up all over the internet, right? It's the middle one, right? We don't really remember the first and the third, but these aren't the droids you're looking for. Hence, it ended up on mugs, greeting cards, names of bands, posters, in the internet memes, right? Pictures of the stormtroopers looking for the droids on Google. This little baby stormtrooper saying, bye, daddy, I hope you find the droids you're looking for. All over the internet, this line appeared, right? So what was special about it? Now, each of these follows its own logic. It's always, very hard to say anything particular. But when you got into some of the machine learning on the text here, you actually found that there were certain recurring patterns to the quotes that went viral from movies. In particular, they had this interesting bifurcation in their behavior. In their sequence of word choices, they were less probable, right? So a phrase like "you had me at hello" from Jerry Maguire has this very unusual bigram, "at hello." We don't normally say that. In fact, that bigram almost never appeared until the movie Jerry Maguire. But in their part of speech composition, they're actually more probable than typical sentences, right? So you notice that "you had me at hello" has the same part of speech sequence as the completely dull sentence, "I met him in Boston," right? And so in some sense, what emerged from that was there is something we can say about the fitness function for text here, which is that it's in a sense a sequence of unusual words built on a scaffolding of common part of speech patterns, right? The common part of speech makes the quote go down very smoothly. The unusual words give it a certain flavoring that makes it stand out. So this notion that we were able to actually take, reconstruction algorithms for transmission, reconstruction algorithms for mutation, ways of estimating relative fitness functions in all these processes were cases where it was very striking over all this time that we were able to actually import these algorithms. I should of course mention that it's hard to look at all this optimization and not talk about the fact that in the years since we've begun to wrestle with the fact that highly optimized social media feeds are not always things that people want, right? They're not always in people's best interests, because in the end we're dealing with something, as social media gets more and more highly optimized, as we use these algorithms for fitness and transmission more and more effectively, we end up latching on to people's self-control problems, right? We end up with a behavioral problem. And so an ongoing line of research at the moment is really to think about all this in the context of one more thing, the consumer, right? And to work towards a behavioral economics of network recommendation algorithms. So that's just a very fast run through of some of the ways in which work from the computational biology community has really had a huge influence on my own thinking, the thinking of my co-authors, as we try working in something which is, again, metaphorically similar, internet information rather than genetic information, but where the algorithms can actually be quite concrete and really lead to some new and specific insights. So again, I'd like to thank you for allowing me to take part in this. Congratulations again, Soren, and it's a pleasure to be able to take part in this event honoring your birthday. Thanks very much.
Thank you so much, John. Such an important topic, and one that I didn't think had any relationship to bioinformatics. So that's really cool. Do you have to run right now, or can you take a question, or do you have to go?
I could take a question, yeah.
Is there one question maybe for John? Okay, I will ask a question. So, so John, the generation of new memes, have you thought of, you have essentially some good priors on what the distribution of bigrams and trigrams should be. Have you thought about maybe a model to generate new memes, or is that too borderline potential for exploitation and evil?
Yeah, it's a good question. So this was certainly something that we talked certainly with people sort of in the early days of language modeling, autocomplete type processes, and that's certainly, I think, a place where you see a lot of this ending up. But I think a lot of the in some sense the sort of accurate modeling of bigram and trigram statistics, and then the notions of like what leads to different human reactions. This is sort of at the heart of the sort of reinforcement learning with human feedback framework, in which I'm taking bigram and trigram modeling of language. I'm then passing that through an objective function that rewards things that people rank highly, downvotes things, downranks things that people don't like, and tries to arrive at this. And I think a lot of the anxieties that you're seeing around large language models, which it goes without saying is one of the topics in computing on everyone's mind at the moment, comes from exactly this question you're asking, that there is the ability to sort of hill climb on the fitness of language, and large language models are arguably more effective at that than any other past technology that we've created. And so I think these questions are going to very much come to the fore as we go ahead with those, and think about those kinds of technologies.
Cool. That's if it's quick. Is it quick? Let's give it a shot. Sure.
Hi. So, in thinking about what made a specific quote special that became viral, did you take into account the initial promoters of those quotes and their scope, the sphere of influence, and so on? And how is that related to biology in any way or form?
Yeah, that's a good question. So we did think about the initial people who spread this in the context of movie quotes, of course, it's a little harder because the movie is sort of the source there. But on the internet, we did some work with groups at Facebook. So Justin Cheng, who I worked on the movie quotes with, moved to Facebook, and we looked at cascade prediction. And there we could take, again, exactly the same meme, look at it start in different places in the network, and then we get to spread. I think there are some interesting evolutionary questions that we didn't really directly relate to any of these particular bioinformatics algorithms, but the question of how different ecological niches can promote different measures of relative success, right? So I look at the same effectively genome in the case of identical internet memes, but I drop them in different points in the environment, right? And certain parts of the environment are more rich and allow it to sort of work better. And there's going to be a codependence, right? There's going to be a coevolution between the content and the location of the network. What works in this part of the network won't work in that part of the network. And so this idea of the fit between the information content and the ecosystem that's being dropped into is very much something that you think about with the spread of memes on social media, as you do in the case of how the genetic information maps or doesn't map onto fitness within this specific environment as opposed to some other environment.
Great, thank you so much. Let's split.
Thanks very much.
So let's move right along. Next we have Ron Shamir, who I assume is on Zoom. Ron Shamir is the Sackler Professor of Bioinformatics at Tel Aviv University. And thanks, Ron, for waiting. I know it's a little bit late for you right now, but we appreciate you staying up for us. Whenever you're ready.
Hello.
Yep, we can hear you.
Okay, can you hold on just a second? I have to restart again. Can you see my screen?
Yep, perfect.
Okay, so hello everybody. Hi Sorin. As some of you may know, Israel was thrown into war today, and we expect tough days ahead, but I didn't want to miss the event, and unless you hear the alarm sound in the background here, I'm with you for the next 10 minutes or so. So congratulations, happy birthday, Sorin. You really had a tremendous contribution to bioinformatics, to creating this field and shaping the community. We heard about it a lot. Let me mention our personal interactions. So we first met when we were both TCS young scientists. And then shortly thereafter, I guess both our interests shifted, and in the DIMAX ComBio program, we met in this field that was at the time still nameless. Then, Sorin, you organized the first ComBio meeting, I think around 94 or 95 in Sandia. Now we call it RECOM 0 because it was sort of a precursor. And then I invited you to the symposium in Tel Aviv University in 95, and you told me about the plans to create this new conference, and my contribution was coining the name, and I'm happy to see that it persisted. And then, of course, we've had tremendous years of RECOM and in creating the community. So really, it was a pleasure to interact and to collaborate. Here is the first picture I found of both of us. This is from RECOM at Tokyo, 2000. We are much younger, much blurrier, and with dark hair. As years pass, you see us again in RECOM around the world. It really took us all over. Beijing, St. Petersburg, and Paris, and the community grows, but you see that Sorin is always in the center. Okay, so I'm going to very briefly tell you about two vignettes relating to personalized medicine. And of course, the vision is that instead of treating everyone with the same therapy, where some are helped, some are unhelped, and few are even damaged, we will use all the data available, medical records, omics, etc., in order to tailor the drug to the individual, and then almost everybody will be helped. So the first story has to do with integrating multiple omics, and this was work of Nimrod, a PhD student in the group who just moved to a postdoc at Stanford. So the data that we are using is the TCGA data, which is really a fantastic resource, tens of thousands of cancer samples. And for each of them, we have multiple layers of omics, mutations, copy numbers, gene expression, methylation, etc. And one difficulty, and each of them was used separately in order to find subtypes of cancer. But when we want to try to use multiple ones together, we see that they are very diverse. And the question of how analyzing together binary beta values, read counts, and discrete clinical parameters is a great challenge. So we first looked at the literature, and we saw that the problem was studied under different names, and there are a variety of algorithms that try to integrate multiple omics data. It even has a name in machine learning, multi-view methods. And we wish to see how they will work on omics data from TCGA. So we took 10 types of cancer with a few hundreds of patients per each and focused on 3 omics: gene expression, DNA methylation, and microRNA expression. And we set out to cluster the data using each of these methods. And in order to evaluate the results, we used the survival function. We knew how much each individual in the dataset survived after the sample was taken. And we looked also at 6 different clinical labels that were available for each of them. But before I tell you the result, I will also mention that we developed our own algorithm, which I don't have time to describe in detail, which is a network-based similarity, where the network is a variant of the radial basis kernel of the k-nearest neighbor network, done separately for each omics and then integrated together. And clustering is a variant of spectral clustering. So here are the results. First, what you see here is the 9 benchmark algorithm and NeMo in the last column. And you see that in terms of significantly different survival between the clusters, NeMo did best. It did best tied with 3 other in terms of the significant number of types of cancer with clinical enrichment. In terms of the sizes, the number of the cluster, you see that it's about midway, not the largest and not among the smallest, and it's a very fast runtime, only faster with spectral clustering. Here is another view of the data. So here you see on the x-axis the average log rank p-value. So better is to the right, and on the y-axis you see the average number of significant clinical parameter, and you see that NeMo is on the Pareto optimal contour along with 2 other algorithms. So since we published this work, there were several benchmarks. Two specifically looked at NeMo together with other algorithms, and they concluded that NeMo did best, in one case jointly with the SNF algorithm. Now, moving on, this is a project led by Jonathan, a master student of the group, together with Nimrod. And here we wish to analyze different omics, but the different omics are for different sets of patients. So unlike the previous problem where we had multiple omics for the same set of patients, here we have this joint set of patients and this joint omics. Can we integrate them together? So specifically, we looked at a situation when one data set was only DNA methylation, and the other data set was only gene expression, and the patient sets are disjoint. So here is a scheme of what we did. So first, we needed to train our algorithm based on data which had both methylation and gene expression for the same set of patients. And then we use regression to extract models for about 2,000 best genes in terms of predicting the expression based on the methylation. And this is done on the training phase. And now in the embedding phase, we have one data set which is only methylation and the other which is only expression, different sets of patients. And we use the models that were built in the training phase to predict the gene expression from the methylation on the first set. And then we clean the set of genes based on differentially expressed genes, both in the methylation and the expression, obtain a smaller adjusted gene set, and then apply canonical coordination analysis in order to form a joint embedding in the same space. And we typically use a dimension of 40 in the reduced space. So here are some results. We looked at TCGA again. We had 11 cancer types with a few hundred samples of each. And in each case, we trained the model to build the regression set on 10 cancer types. And we predict the gene expression on the 11th. And then we do the embedding of the predicted and the true gene expression. And since we know in this case what is the true match between the samples, which embedded gene expression and predicted one come from the same patient, we can measure the quality of the result. So what fraction of samples are closer than the true match? So here are the results, 11 cancer types and 7 or 8 algorithms. The green one on the left is intent. This is the name of our algorithm. And you see that it is by far the best in all cases, typically 1 or 2 orders of magnitude better than all the other algorithms. Here is another view. This is looking specifically at colon adenocarcinoma. You see the mixing that is obtained by the intent algorithm compared to the much poorer mixing by the other 3 algorithms that we tried. One more challenge here, we wanted to see if we can integrate multiple cancer types together. So we looked at 4 cancer types. We trained the model on the remaining 7 types. And then we do the embedding again. The score that I described before is again 2 orders of magnitude better, but here we also want to look at, so the second row shows us the mixing, and we see again that the mixing of intent is very good, but the third row shows the differentiation between the 4 cancer types. So you see that Intend and in this case Seurat are doing a pretty good job in terms of separating the cancer type, and only Intend does well both in terms of the mixing and in separating the cancer types. So I know that Sorin asked for an open problem, so here is my open problem. Devise an effective integration method for this time for any kind, of omics. What we did was specifically for two kinds where we knew the relation between them, but the general question is still open. So in summary, we described two algorithms, one for multi-omic clustering and one for integration and two specific types of omics from disjoint cohorts. And both algorithms and the software are available from our group website. I thank my group for everything and congratulations again, Sorin. I'm going to join you at 70 next month, so I look forward to being with you there again. Bye-bye.
Thank you so much, Ron. Any questions for Ron? We have time for like one. Oh, we have one. For Tambra.
Excellent talk. Thank you so much. I guess my question was that some of the baselines that you compared your model with are very sensitive in terms of the hyperparameters you select. Like their results are very sensitive to that. I wonder if you had any thoughts of seeing that, what kind of exploration you did in terms of that, and how does your model compare?
Yeah, so it's a good point. We followed the recommendations for hyperparameter choice in each of the algorithms in the benchmark. And so indeed, one can do a more extensive parameter search, but some of these algorithms were already quite compute intensive, so we just followed the recommendations in the original paper.
Let us thank Ron again. Thank you so much, Ron.
Thank you.
So up next, we have another Zoom talk. We have Moshe Vardi. He is the University Professor, Karen Ostrom George Distinguished Service Professor in Computational Engineering at Rice University. Looks like we can see your screen.
Okay, and you can hear me?
And we can hear you well. So whenever you're ready.
Okay. Well, let me start by congratulating Sorin on this auspicious occasion. I would have loved to be there in person, but life intervened. Sorin had a great career. There was only one blemish on his career. We made him an offer to come to Rice, and he declined our offer. One of my life regrets. But he's done well nevertheless. I want to change tone a little bit and maybe kind of follow up on John Kleinberg's talk, but looking not at the internet technically, but looking at how the internet has affected society. And this is kind of a bit of a clickbait, how to be an ethical computer scientist. I will not really give you a manual, but I want to start a conversation. And I want to point out how the previous decade has kind of changed things and how we think of computing. So just looking at ACM, where I've been very active, in 2012, we celebrated Turing Centenary, which is all a celebration of the technology. And 5 years later was the Turing Award at 50, another celebration of how ascendant we have become. And then in 2022, just a bit over a year ago, ACM celebrated its 75th anniversary. And now the tone was a bit more sober and somber. Panelists will imagine what might be next for technology and society in the years to come. And many of the talks look at societal impact. So much so that one fairly distinguished participant, who will remain nameless, posted on Facebook, I found the whole thing a little depressing. Why can we not just not celebrate the technology? But you cannot divorce what happened in computing, what happened in society overall. Notice the date, June 10, 2022. One day before that, the House of Representatives started its hearing about January 6th insurrection, where we learn how essentially the President of the United States tried to subvert the legal transfer of power, the lawful transfer of power. And 2 weeks before that, here in Texas, we had the Uvalde massacre, where young gunmen killed 19 students and two teachers in a very, very tragic event. But I'm mentioning these 2 events because they point to a very, very deep polarization in American society. In July of 2022, New York Times held a poll about Trump. 92% of Democrats think he's threatened U.S. democracy, but 76% of Republicans think it was exercising his rights to contest the election. Regarding assault type weapons, AR-15, among gun owners, NPR survey in June 2022, among Democratic gun owners, 84% are in favor of banning the sale of such weapons. Among Republican gun owners, 75% object to such a ban. When you have such polarization, it leads to political paralysis and it puts democracy in danger because it erodes trust. Democracy requires a basic trust that we are all in it together. And this is being gradually eroded, so much so that people are seriously talking about the disunited states. You know, people are analyzing the possibility of the United States breaking into red and blue confederacies. You know, it's speculative, but the fact that people are speculating tells us something. So how did we get here? What happened? So for this, I'm going to take us back to 1981, where 3 pivotal events happened. The first pivotal event is that I arrived at Stanford as a postdoc on September 6th. Well, maybe it was not so pivotal, but about a month before that, IBM introduced the IBM personal computer model 5150, which nobody remembers, but it was the IBM PC. And I'll come back to the IBM PC in a minute. And earlier that year, Ronald Reagan was inaugurated as US president. So let's look at this impact of these 2 things, IBM PC and Ronald Reagan. So the IBM PC was a smashing success. By January of 1983, a bit more than a year after it was introduced, Time magazine, usually it has the person of the year on its cover. Now it has the machine of the year. The computer move in and you see a desktop and a man looking rather glumly, in my opinion, at the desktop. And the IBM PC launched a tsunami of technology. By mid-80s, we had a computer on the desk of every knowledge worker. By the late 80s, every knowledge worker took a computer home. The World Wide Web was introduced in 89. Internet became commercial in 1995. Google was launched in 98. Facebook in 2004. iPhone 2007. And now each one of us is carrying a supercomputer in our pocket, a computer that's more powerful than the computer that sends Apollo to the moon. But we must also not just focus on technology, but look at the socioeconomic context. Ronald Reagan launched a tsunami of neoliberalism, which is extreme, some people call it free market fundamentalism, which include basically minimize the states and just give power to the free market. So economic liberalization, reduction in government spending, privatization, deregulation, monetarism, tax reduction, globalization, free trade, altogether by 2006, Warren Buffett said there is a class warfare, all right, but it's my class, the rich class, that is making war, and we are winning. And what was he talking about? He talks about the dramatic increase in economic inequality, economic polarization in the United States. What you see here is just comparing the 1%ers to the 50%ers. So if you go back to 1962, the 1%er had about 12% of the income, and the 50%er, the bottom 50%, has about close to 20% of the income. And then you go over a period of over 40 years, 50 years, and you see it mostly starting around 1980, you see that we have a reversal. The bottom 50% has gone down to about 12, 13%, and the 1% has gone to about 20% of the income. So we have great economic polarization in this country. Now, this economic polarization was amplified by cognitive polarization. And it goes back to again, Ronald Reagan. Under the Ronald Reagan administration, the fairness doctrine was abolished. What was the fairness doctrine? The idea was that any broadcast license holder, ABC, CBS, NBC, must represent the news fairly. So reality is, it didn't matter if you watch ABC or CBS or NBC. It was more preference which anchorman, they're all men, which anchorman you liked, and but they all represented the news in a balanced way. Now the FCC then decided we don't need anymore because we have cable channels, so people can get all point of view by watching different cables. But people don't watch different cable channels. They choose one channel, that's what they watch. So the people who watch Fox News and the people who watch MSNBC are living in different realities. That's what we call today cognitive filter bubbles. And then came the internet and amplified it, because when the fundamental business model for the internet was advertising. People discovered that just standard advertising did not work on the internet, and Google invented micro-targeted advertising. So we match the ads to personal preferences, and that required lots of personal data. So this became known as surveillance capitalism. And basically right now, everything you see on the internet, unless you go to www.cs.rice.edu/tillavardi, everything you search, everything you see on social media is really mediated by machine learning. And so we got, again, filter bubbles. We live in different realities. And this got us to where this country is. And I feel myself today as Enders. This is from the science fiction novel Ender's Game from 1985 by Orson Scott Card. And spoiler alert, in that book, Ender is a teenager. He and his team they think they're playing video games. They're not realizing they're actually fighting intergalactic war. And when they win the war, they've destroyed the civilization. And that's where they end up. Oh my goodness, what have we done? And that's how I feel today. I got into computer science because it was fun. And now I look, I say, oh my God, what have we done? We thought we were the Rebel Alliance, turn out we are the empire. So the ACM Code of Ethics starts with the following paragraph. Computing professional actions change the world. To act responsibly, they should reflect upon the wider impact of their work, consistently supporting the public good. So how to be an ethical computer scientist? Consistently support the public good. And when we teach this at Rice, this is a quote from some Rice student where he explained why he will not go to work for Facebook. He said, I'll have a bachelor's degree from Rice. My skill will be in high demand. It will be easy for me to choose a company that is not ethically questionable. You don't even have to say that Facebook is evil. It's enough that it is questionable. I can go to other companies. So just finishing with reflection on us as CS faculty, if we educate socially irresponsible students, then we are not supporting the public good. So it's our social responsibility to convey this value of social responsibility to our students. It must be part of the CS curriculum. And I encourage you to look at the paper that I wrote with a colleague at Rice about 2 years ago on deep tech ethics. Thank you very much.
Questions?
How do you decide whose view of social responsibility to use?
If you read the US Constitution, the first paragraph talks about the public good. Just go read the preamble to the Constitution. It basically said the defense, a common defense, etc., more perfect union. And then there is a question, again, how do we as a society decide what is the public good? And the rest of the Constitution gives you a mechanism how we go about to decide the public good. And the answer is, it's going to be hard. We all know democracy is hard. It's the worst governance system except for all the rest. So there's no magical way by which I tell you there is an oracle who will tell you what's the public good. It's something that we are negotiating and discussing and debating, and I don't think we'll stop doing it. But we must start this conversation. You can look at some things that we are doing, and maybe they are debatable, On the other hand, you can look at some behavior by some tech companies, and the answer would be, I think it would be very easy for us deciding that Uber basically misleading the government is not a good thing. It doesn't serve the public good. So, but the answer is lots of debate, lots of discussions.
Any other questions? Yeah, a quick one maybe. So how do we become the Rebel Alliance? What sort of changes do we need to make to social media platforms or anonymity on the internet or incentive structures to fix things, or at least bring things closer to normal?
So there are many, this is now how to regulate technology active of intense debate all the way it's finally made it to Washington, D.C. I have my proposal how to start things. And the answer is, whenever you use technology, you live in a shadow legal system. You don't live in a normal legal system where the laws are passed by Congress and signed by the president. You live in the shadow legal system where all the licenses are written by tech companies that are meant not to be read, and they're not read. New York Times calculated if you want to read all the licenses for technology used in a given year, you'll spend 2.5 months just reading these licenses. And so the answer is, we first have to eliminate the shadow legal system. I would advocate a simple law that says all click-through licenses, past, present, and future, are hereby nullified. Now, do we need law for cyberspace? Absolutely. But again, let's use democracy to establish these laws, rather than let big tech corporate lawyers write these laws.
Great. Oh, we have one more question from Bill.
I'm sorry.
Oh, no, please.
At first, I thought you were saying something you weren't. Then I decided it was because English was your second language. You said we should not educate socially irresponsible students. I think you meant we should not turn out socially irresponsible students.
Yes, yes.
And if you didn't, then I worry about the issue of social responsibility against personal freedom really scares me. I'm an advocate of Hayek. I'm sure you're familiar with Hayek. And I wonder if a young student who is also an advocate of Hayek would in fact be considered not worth educating.
So I think you should read Hayek a bit more carefully. For example, people think of Hayek advocated no state involvement in the markets. It's just not true. Go read Hayek. I'm happy to send you references. Hayek was definitely scorned Van Mises, who was even more extreme than Hayek. Okay, so go read. I would suggest that you read Hayek and open your eyes a little bit to what he's actually saying.
So what I'm worried about is I'm listening to you tell me, oh, I shouldn't believe in Hayek. Well, fine, that's good.
No, I didn't say that. I said go read Hayek more carefully.
I have read him quite carefully, but that's not my point. Even if I'm wrong, that does not mean that I should be denied an education.
No, I did not say that the students who believe in Hayek should be denied education. I think you should discuss with your students social responsibility, and there will be diversity of opinion, which you should welcome. I don't think all the students are required to adhere to my view of social responsibility, but they should understand, we take other concept. We take the concept of justice. I assume you believe in justice, but we debate what justice means.
Certainly.
Turn out 70% of the people, 70% of the people in this country believe in social justice. Of course, there are huge differences how they interpret it, and that's the way it should be. But let's start the conversation with our students.
May I say one more thing?
Sure.
My only worry is that we're seeing a tendency in this country right now for one side's definition of social responsibility to shut out any discussion from the other side of the issue. And I'll let it go at
Yeah,
that.
we can leave it at that. I don't think it's not something that Dr. Vardy was purporting, but that is, I think, a phenomenon that does exist in many different areas. So, which is not healthy for a conversation. So let's thank Dr. Vardy again. Thank you so much. We have to move along. We have ended the Zoom part of this program this morning. So we're going to move on to Dr. Peter Shor, who is the Moores Professor of Applied Mathematics at MIT. and winner of the Gödel Prize and MacArthur Fellowship. Dr. Shore, please come on down. Do you have your own computer? Sure.
So you're not going to get a break?
I know, we had a break, but then we had a, we can have a 5-minute, we can do a 5-minute break while we set up and stuff.
Yeah,
you know.
okay.
How are you?
Good.
Take 2. It's my pleasure to introduce Peter Shore. Peter Shore is the Morris Professor of Applied Mathematics at MIT and winner of the Gödel Prize and MacArthur Fellowship. Peter, I'm looking forward to your talk.
All right, thank you. So I'm going to talk about there's missing okay, I see. So I'm going to talk about some work I did maybe 10 years ago on on statistical mechanics in quantum systems, and there's a very related, very interesting phase transition, and I will, mention that at the end. So I'm going to be talking about the Motzkin and Fredkin quantum spin chains, mainly about the Motzkin spin chain. I didn't realize that I would need to throw almost all the material about the Fredkin spin chain out of this talk when I gave them the title. So what is a spin chain? A one-dimensional quantum spin chain is a one-dimensional array of binary dimensional quantum systems. So for example, in this talk, these systems will each have 3 dimensions, and they're connected by a local Hamiltonian, which gives the energy. And each quantum system is a vector space. And in this case, it's a qubit, so it has 3 dimensions, and then there are 3 basis states, which we'll call, for the time being, A, B, and C. And so what happens when you look at the whole quantum system as a state space? Well, what happens is now you have a state space of size 3 to the n, or dimension 3, complex dimension 3 to the n, and it's the tensor product of each of the individual state spaces. And 3 to the n is a huge number, and that makes simulating this system very difficult. And the basis states of the 3 to the n dimensional system are, just the tensor product of the individual basis states, which we will abbreviate as AC. Instead of saying A, tensor C, tensor A, tensor B, we'll just say ACAB, to make the notation simpler. And one really interesting thing about spin chains that people look at is what is the ground state? And now I want to say a little bit about entanglement. Suppose you have a quantum system with 2 different subsystems, A and B. And in this case, the 2 subsystems are going to be the first half of these spins and the second half of these spins. And the entanglement entropy measures how much quantum correlation there is between A and B. So quantum correlation is related to the spooky action at a distance. And if you look at A, then it makes B change in some very subtle way that is not allowed to transmit information. And you can quantify the entanglement entropy fairly easily. And this is the formula. It's the entropy of the eigenvalues of the partial trace of psi psi. And if that doesn't make any sense, you don't need it for the talk. You just need to know that it measures quantum correlation between the 2 sides of the subsystem. Now, when we started this research, there was this conjecture called the area law. And it says, for the ground state of a quantum spin chain, the entanglement entropy across any cut is proportional to the length of this boundary. And it's been proved for one-dimensional spin chains with constant spectral gap. So the constant spectral gap means that the minimum excited state has a constant energy above the ground state. So the difference between the first excited state and the ground state is a constant energy, where the energy is expressed as a proportion of the natural energy level of these individual Hamiltonians on adjacent qubits. Okay. So it has been proved one-dimensional spin chains with constant spectral gap. And that says, in a specializing to one dimension, that says if you have a one-dimensional spin chain that has a constant spectral gap, and you make a cut, then there's at most one bit of entanglement between the left half and the right half. And it's still a conjecture for higher dimensions. There are no counterexamples. So what the Motzkin and Fredkin spin chains showed that the spectral gap assumption was necessary. And actually there had been previous papers showing that this assumption was necessary, but they worked quite differently from our paper in that the previous papers basically showed that you could violate the assumption by building a universal computer that worked on the ground level of these energy systems, and the universal computer could give you any energy gap you wanted. But here, these are much, the Motzkin and Fredkin spin chains are much simpler than these universal spin chains. And, they show the same thing. So what it is a spectral gap is 1 over n to some constant. And this constant is 3, probably, if you believe in simulations. And the entanglement between the left half and the right half is either 1/2 log 2n, or if you go to a, more complicated related spin chain, it's order of root n. And if you go to an even more complicated related spin chain, it's order of n. So what are these spin chains? So I'm going to have to tell you what a Motzkin walk is. And this is purely classical. A Motzkin walk is a walk from 0, 0 to 12, 0. And each of whose steps is either 1, 1, or plus 1, or plus 1, 0, or plus 1, minus 1. So they're either up 1 step or level or down 1 step. So here's a Motzkin walk. And the other 2 things you need to know is that it starts and ends at 0 height, and it always stays non-negative. And what do we need to know about Motzkin walks? Well, there are some rules here. You can change this state up followed by a level 1 to a level 1 followed by up. So here's an example where we've done that. And if you, and level can go to up, down, and up, down can go to up, down level can go to level, down. But I want to note that I do not have the move up, down to down, up. And by using these moves, you can go from any Motzkin walk to any other. Now, if we had the move level to down, up, it would take us away from Motzkin walks, because we might have a level, level at, height 0, and then that would go down, up, and that would give you a height -1, and we don't want that. But these moves never take you out of the set of Motzkin walks, and you look at this this Markov chain where you choose a random one of these moves and make it. It will eventually take you to any other Motzkin walk, and we can find the mixing time and the associated quantities. So what is our Hamiltonian for a Motzkin spin chain? Well, we will have special terms at the left and right ends, which I will mention later, but the Hamiltonian for everything between spin I and spin I plus 1 is level up can go to minus up level, and level down minus down level, and up down minus level. And what this means is that so each of these spins is either a level step or a down step or an up step. And what this Hamiltonian means is that the amplitude on up, if you take a Motzkin walk, it's one of the elements of the superposition in the Motzkin spin chain. And what this means is that the amplitude on the up, on a Motzkin walk with an up level step, is equal to the amplitude on the Motzkin walk with a level up step in the same position. Because, if you have up level minus level up, that ends up 0. So if these two amplitudes are 0, then there's no energy from this term. But if they're different, you get an energy contribution from this term. So it's not in the ground state, because we will show that the ground state has 0 energy. And the other two things is we had add a penalty term for a down step at the left of the chain and up step at the right, which means that basically it will always be bigger than 0. So the only 0 energy ground state is a superposition of all Motzkin walks. And I should have put a normalization term in it here. Okay. So if the only 0 energy of the ground state is summation is the superposition over all Motzkin walks normalized to be a unit vector, what happens when you look at the dividing line, the correlation between the left half and the right half? Well, you know that the middle of the walk is at some point, maybe that's 3 steps up, and then you have to go 3 steps down in the right-hand side of the walk. So this is a correlation between the 2 different sides. And there are n possible values of the correlation, and the average value of the correlation is the average value of the number of steps that goes up is root n. So that means that the entanglement between these 2 sides is 1/2 of log of square root of n, or rather it's log of square root of n, which is 1/2 log n. Okay, so we got 1/2 log n bits of entanglement here. Now, what you can do now, you can refine this and write your Hamiltonian so that if you have a step that goes up, so now you have 2 different colors of the up steps and down steps. And if you have a step that goes up and you look to the right, the matching step that goes down has to be blue. And you do that by saying that if you have 2 blue steps in a row, 2 blue things in a row, they can go to 2 flat steps. But if you have a left step, if you have an up step with a red and a down step with a blue, then they cost you energy. And that means that, what that means is that the ground state is made up of a superposition of Motzkin walks with matching colors. And now you can see that the sequence of steps here, red, red on the left, has to match the sequence of steps down, right, red, red on the right. So we have, 2 to the n bits that are matched between the left and the right. And this gives you a log, or rather, log of 2 to the n, which is, or log 2 to the root n, which is root n entanglement between the left half and the right half. And actually, you can get more than that. And you can introduce a phase transition. And here what you do is you put a weight on these terms in the Hamiltonian, which favor steps up and disfavor steps down. So what you get is you get you're going to have to get the each of these Motzkin walks in the term is weighted by the area of the area underneath it. And so if t is less than 1, your walks will on average be flat. And if t is greater than 1, your walks will probably go up to height order of n and then come back down. And at t equals 1, that's the previous Hamiltonian. That gives you the peak at square root of n of the height of the Motzkin walk. And so this is a phase transition between order of 1 entanglement and a gap, a quantum system with a spectral gap, and quantum system with order of t entanglement between the left and the right, and a 1 over n to the c spectral gap. And you can ask, what are the properties of this phase transition? And I want to say, I don't think we know. I mean, so I have not looked at the follow-up papers to the paper which gave this phase transition, which was by Zhang et al., but I haven't seen any in the last, this wasn't so long ago, so I haven't seen it in the last 5 or 6 years. And now, there's also the Fredkin spin chain, which I mentioned in the introduction. And the main difference is it's Dicke walks, so there are no level steps either up or down, and the moves are more complicated. The Hamiltonian is defined on 3 adjacent spins rather than 2, and the moves are up, down, up, goes to up, up, down, and down, up, down, goes to up, down, down. And if you do this, then the properties of the Fredkin spin chain are very similar to that one. And I have one last open question in my talk, as Soren introduced it. So the question is, what happens to the limit as the number of steps in your chain, in your spin chain, goes to infinity? Well, what happens is if you scale everything properly, the state becomes a superposition of Brownian excursions. And a Brownian excursion is just Brownian motion constrained to be 0 at the first point, 0 at the last point, and never going below 0. Now, if you take the limit of many spin chains as n goes to infinity, what you get is a conformal field theory. So what we can show is that this does not go to a conformal field theory because, none of the scaling quantities or energy or anything else is right for a conformal field theory. But what does it go to? You assume that there's some nice description of the limit, but as far as I know, nobody has found this. So that's my open question, and thank you for listening.
Questions, please.
So I know you would like an analytical theory, but there are exact results, close to exact results that Hanson matrix normalization theory can give you in 1 dimension. So how much insight can you get from that? DMRG scales pretty well in 1D, hundreds of sites.
Yeah, so DMRG actually does give you a lot of things about these original Motzkin spin chains. And what I don't know that DMRG does is give you anything, tell you anything about this limit. But I don't know if anybody has looked at DMRG and tried to use it to analyze the limits, so it's possible that that might be the right method of attack.
Any other questions?
As a biologist, I almost hate to ask you any question whatsoever. I've tried to understand entanglement. Social entanglement has no limit on its distance. Is there a physical limitation to entanglement?
That's a very good question. So generally, if you don't put any restrictions on the system you're dealing with, there's no physical limit to entanglement. if you look at quantum spin chains, there is actually a physical limit to entanglement. If you believe in the area law, where was the area law? And I believe that the physical limit to entanglement is something like 1 over the spectral gap. So if you have a constant spectral gap, yeah, I think that's right. There's some function of the spectral gap which gives you the physical limit of entanglement in a spin chain.
Does this agree with the experimental data?
It's very hard to measure with entanglement and spin chains with experimental data. People have done experiments where you have entanglement measured over kilometers. So, and you have entanglement between the Earth and a satellite up there. So it's certainly possible to get entanglement over very large scales.
Thank you so much. So lunch is going to start a little bit later, and it'll end later. So the second session will start a little bit later in the day. We have 2 more talks. Give your own, yeah. Yeah, yep. Okay. Oh, Dr. Short? Oh, sure. Thank you. So next, we have Bjarne Stroustrup, Professor of Computer Science at Columbia University, winner of the Faraday Medal, and designer, inventor of this language you might have heard of, C++. So we're getting set up now. Yes, take it away.
It worked. Wow.
Perfect.
So here we're going from science to engineering. Engineering may be less glamorous, but it's essential. You can't have stuff out on L2 without some good engineering. And I am convinced that every one of us has used something built on C++ several times today. Anyway, I'm not going to go deep into anything. I will show some code because I once promised myself I would never give a talk without showing code, because that would be inappropriate for somebody talking about languages. But there's some interesting things going on here. Basically, for me, it all started with a problem. In my opinion, most good tools, most good languages starts with a tool. And I had the ambition, I wanted to build a distributed Unix back in the late 70s. If I had succeeded, I would have beaten the records with 10 or 20 years, but I got distracted. No language at the time could do what I wanted. I needed raw performance, and I wanted to manipulate hardware, and I wanted to abstract from this messy low-level stuff to specify how 2 pieces of software on 2 different machines, maybe, or maybe not, could talk to each other. That needed abstraction, it needed formalization, it needed good, solid, checked rules. No language could do both. There was languages that could do either. So Simula could do the abstraction to some extent, and C could do the hardware manipulation. So obviously, we just merge these 2 and get it done. It couldn't be done, but nobody has told me that, so I did it. Anyway, so basically C++ rests on 2 pillars, a direct map to hardware. Even unusual hardware, you can compile or work your way down to FPGAs and GPUs. The ideas came initially from C, and zero overhead abstraction in production code. I want to abstract, and I want to be able to afford to abstract. I mean, it's no good that you can just do abstract things if your computer can't run it at scale. It initially came from Simula, which was too slow, but I fixed this. And ideals must be articulated. This is a problem I've had over the years. I try to explain what this is about, and people aren't listening, or I can't explain it to not. And people don't like philosophy. They want tools. They want something they could use yesterday, preferably. Oh dear, so we have to talk about that, and it takes time. So another way of looking at it is C++ comes from two solid traditions of computer science. There's a hardware-focused thing where you want to look for every byte in every cycle and get it optimal. Starts with assembler. That's David Wheeler, who happens to be my thesis advisor, and writing the first program on a modern stored program computer in '49. This is a very young field we are in. And anyway, BCPLC, and I borrowed it, that's Dennis Ritchie. He was my colleague for about 16 years. And here we've got Bacchus, who invented Fortran, which was the first language that was sort of built to be good for humans, instead of looking at the hardware. Very peculiar kinds of humans, of course, engineers and physicists, but fine. And once we got Fortran, everybody wanted their own, COBOL, et cetera, et cetera. And here comes Christian Nygaard, which says, why do we build all of these different languages so people can't talk to each other? Let's have a way of making abstractions in general. And that's where we get classes, class hierarchies, object-oriented programming from. I met him in Aarhus too. So anyway, from there came C++, and from there came a lot of other things. That was the historical view. But anyway, the key idea here is to represent ideas in code. I don't actually want to have things on my whiteboard in my head and then being some kind of compiler to turn it into bits and bytes and addresses and such. I want the language to do that for me. And so every field has some ideals, some concepts that you can put into code and has been put into code. And there are just lots and lots and lots of them. And then you have to make it affordable. As I said, I couldn't afford big computers. I was a grad student. I was a young researcher at the time. And so I've always looked at sort of economy. And there's two areas today where this kind of thing is really, really important. If you have huge server farms, if your language is twice as slow as something else, you need another one. And you just there blew $60 million, $100 million. The other one is sort of wristwatches and things where you run out of battery if you're slow. So your imagination is the limit here. And one of the things that sometimes annoys me is that you can never see it. If it's done well, it's basically not there in people's minds. So if it's done well, it's invisible. And so I'm going to show you some code. Here is a classical bad program. I open a file, I use it, and I close the file. This is a classical source of bugs, because all the compiler sees and all the programmer that doesn't know this sees is there's a pointer coming out of a function, and we use it, and we have to remember to close it. It says so in the manual, and nobody reads the manual. And so you put a return statement in here, or throw an exception, or in C, do a long jump. You never get to there, and you have lost something. It used to be that most of the browsers were losing images that way, and eventually freezing. Classical bugs. So what's wrong with this? I said you had to represent your ideas in code. There's an idea of a file or a file handle here, but it's not really in the code. I mean, you can understand the English maybe, but that's just a name. So what we do, we represent the notion of a file in code. So I built a little class like this, and I said, well, to get a file handle, you have to give a name and some access thing, and it'll open it. And if it can't open it, it gets out of there without making it. Once you've made it, eventually you have to clean it up again. You have to close that file or your system will go down. That's what that thing is for. Constructor, destructor. That's a bad pun, but I could never resist them. And it closes it up. So you can now write your code. Give me a file handle to a file called S with read permissions and use it. And however you get out of that scope, you fire this one and things are closed. And this works for all kinds of resources. Let me see, did I have it here? Files are a simple example. Memory, people talk about memory handle. Locks, if you have basic system, transitions, communication channels, GUI connections, threads, shaders. It's such a fundamental thing that we grab a resource, we have to give it back again. Giving it back again is not easy. It sounds easy, but ask any librarian. Books don't come back again by themselves. We forget. And so we make these kinds of mistakes again and again. Okay, so basically focus when you come to design on use and users. A lot of talk in computer science and research is all about the techniques and the implementation details and such. Anyway, let's look at this and say what you want to be done, not how it should be done at a level, and you can optimize again later. We have layers, abstractions, interfaces, type safety, and all the good stuff like this. By the way, everything comes out of an environment, out of a culture. And this is where C++ came from. Bell Labs, Murray Hill, one of the greatest places on earth at the time to work. We got hardware stuff that's over in building 1, software stuff in building 2, and there were some great people, huge range of interesting problems, always somebody who knew something that could help you. And basically the rule of thumb was innovation is invention plus development. You can't just have an idea, you have to develop it into something useful. Well, and if you don't have good ideas, you can't build anything useful. And so all good engineering relies on feedback. Somebody says, why didn't you just get it right the first time? Well, I couldn't. I didn't even know what right was. You have to have some good ideas and you have to develop it. So you have to deal with the changing world, deal with this, and you have to rely on the feedback. And so people want a simple language. Actually, a request I get at least twice a month. C++ is too complicated. You must simplify it. And while you do it, I want these 2 features. They're absolutely essential. And whatever you do, don't break my code. I've got 1 million lines of it. This cannot be done. This is a trilemma. What you can do is you can simplify the use of the language, but you can't simplify the language itself without really pissing off hundreds of thousands of people. And that's not such a good idea. Interesting thing, see the engineer there, the cylinder head number 5? This is a large engine. It's been running with its fuel injection implemented by some friends of mine. So anyway, this is sort of interesting. Why do we evolve? People say, why don't we just stop and do something else? And the point is that the world changes, we learn how to do things. Yeah, C++ was, when this was a telephone, this is a wire you put into the wall. This is to scale, a more modern version of the same idea, a supercomputer as was mentioned. And I don't believe in perfect languages. I believe in evolution to meet challenges. And anyway, as I said, this talk is about how to do something useful. Even I could design a prettier language. That's not hard. It is hard to get something that is really useful and convincing people that it is. And so I was doing things like generic programming. We don't just want a vector of doubles. The physicists can do that. That's all they need. But vectors of just anything. And I want algorithms running over it. I want a sort function that can do anything over a sortable range. And so we can have vectors of different types and just ask to sort them. This was not in C, it was not in Simula, it was not in C++ till 10 years later, but we learn, and we learn from the needs of the users. And one thing we have to remember, code is written by people, for people, and it's a tool. people can get obsessed with the beauty of their tools and the theories behind them and such. I'm still serving the users. It's one part of a technical community. It's one part of tool chains. And so I said here, C++ is a general purpose programming language designed to make programming more enjoyable for serious programmers. That is the opening statement of C++ programming from '85. The editors removed it either 2 or 3 times because that couldn't be it. You have to write about reliability, performance, reuse and such. Nah, it's about how to write good things. By serious programmers, I mean people who build systems for other people. I wouldn't say professionals because some of the best are really amateurs. They love it, they write it just because they feel like it. And anyway, it's human. There's humans involved everywhere. And basically it sort of worked. C++ grew, people complain about that, but the usage also grew. And so there's a lot of it. Some of it I'm proud of, some of it I'm not. I mostly picked my pictures of things that aren't socially unpleasant. You heard Moshe. I think about that a lot, but this is not what this talk is about. Thank you.
Thank you so much, Bernie. Questions?
Is there anything fundamentally different about the modern artificial intelligence, the languages that support those systems, or is it just, built on what you're talking about here?
There's so many languages, and there's new languages coming all the time. And I don't see anything really fundamentally new. One of the things that I find with languages in general is that people come up and they will be the world's best at 1 or 2 things. But to be a useful tool, you have to be good enough at everything that your users need. And new languages tend not to have gone through the grueling process of being used in many, many fields. And so that's the strength of an older language. Newer languages are simpler and usually noticeably better at something or other. Then we learn from them. And but I don't see any really brilliant new ideas today. People talk about AI. Well, TensorFlow is a C++ program. We're there.
So is it a desirable property of future languages that they should be most able to be well written by ChatGPT and such?
No, I'm not a fan of that kind of AI. And I really want critical stuff to be written by engineers. There is a problem with education in programming and computer science, is a lot of people treat programming as everybody should write code. And languages should be, and tools should be designed so that everybody can write code. It doesn't scale, it isn't reliable. I really want a different way of teaching scientists and engineers and everybody. I do not want everybody to program the brakes of my car if I had one.
Modern C++ is very highly abstracted, of course, and if you make even a simple mistake with maybe the standard library, you can get errors with templates upon templates, and it can be hard to understand. What are your thoughts on that problem of, what are the limits of abstraction that you think is right?
Yeah, I've been addressing this for a while. The first thing is that most of the complicated template code is there because people compute values, perfectly ordinary values. Don't do that with templates like that. Do a const export function. C++ is really good at calculating values. I mean, floating point numbers, dates and such at compile time. If that's what you want, do that. Secondly, concepts is the precise specification of interfaces to classes. And that addresses directly the problem of bad error messages. We still have to have the compiler writers catch up with that, but the mechanism is there. And then finally, I have to try and deal with the fact that C++ has a lot of barnacles from its history. And I try to do that by separating rules for use for rules for the language. And the rules for use, my work is in something called the C++ core guidelines that can be enforced by tools so that you can write a program and ask it to be checked for certain things you're interested in, and it will stop you from getting into the worst messes and the lower level sort of C-style code with void stars and other nonsense.
Cool. Thank you so much. So next I'll ask Brenda Rubenstein to come up and just give a quick introduction. So Brenda is an associate professor of chemistry and physics. Yeah. Brenda Rubenstein is an associate professor of chemistry and physics here at Brown University.
Good afternoon, everyone. It's my great pleasure to introduce Ken Dill to all of you today. Many of you come from a computational biology background where you think a lot about how do you infer different processes from sequence. But many of us, including myself, think a lot about the molecules in between. We want to understand their shapes, what functions they perform, and in great detail, sort of the atomistic mechanisms behind many of the processes that underlie biology. Ken Dill, beside me here today, who's the director of the Laufer Center for Physical and Quantitative Biology, is really one of the key people who made what I'll call the biological chemical sciences theoretically sound. So Ken Dill was one of the first people who really thought about how proteins fold and come to the shapes that they do. So you may not know historically, but for many years it was sort of posited that proteins could potentially fold in an infinite number of ways. They can access large swaths of conformation space, and people were unsure, how does a protein actually come to its typical native state, or maybe a few key conformations that it might access in time. You can imagine, sampling this huge space, how is it that a protein really settles on one specific conformation? During his early days, Ken was one of the first people to really think about this in a true statistical way. So what Ken realized is that proteins have ways of sort of agglomerating step by step down a funnel, a free energy funnel, for those of you who are familiar with free energy, until they reach their free energy minima. And so Ken was really the person who made us all realize how proteins truly fold down a funnel as opposed to a tunnel, which allowed us to truly understand protein physics. Ken has done a number of things in his life beyond that. He's really thought about protein folding in a variety of different ways, but also thought about different types of life and how statistical physics affects life. For those reasons, he was recently awarded and honored as part of the National Academy of Science, and also he's a member of the American Academies of Science. And so with that, I'll let Ken tell you a little bit about, I think, protein folding and the evolution of life.
Thanks for that kind introduction. So I want to start first by telling Soren happy birthday. And I have this little modest offering, just a little quote, and this reflects my view of how Soren thinks about the world, how a lot of you may think about the world as well, but it's something that I value enormously, and in biology we often don't. And that is, this is a Peebles quote, nature occasionally operates by rules we can discover by pure thought. So I want to tell you about the origin of life. Now this is a sort of third rail of science. You kind of don't want to do this, especially if you're a young faculty member. You'll never get funding. There's no experiments. Nothing's ever walked off a lab bench before, with perhaps a few exceptions. And there's been lots of speculation. Books are written about this sort of thing. So I approach this talk with a great deal of humility. But we have two ideas that drive us to think it's worth rethinking this problem a little bit. And I'm going to mention what the driving forces are. We're thinking about this a little differently. And that is, so life started 3.5 billion years ago. Evolution wasn't running yet. Darwinian evolution doesn't work on chemistry and physics. Darwinian evolution works on biology. So evolution must have had a beginning about then. And the beginning of evolution is the beginning of a process. And we have to think about not just what molecules we might like as the beginnings of life, RNA or proteins or lipids or what have you, but also how a process could start. And by thinking about, you might say sort of this breaks down into thinking about the nouns and the verbs. A lot of origins of life work thinks about the nouns. That is to say, was it RNA, was it proteins, was it lipids? We need to think about the verbs. We need to think about what's the process, what's the action, what's happening, why does the thing keep rolling? And when you think about the verbs, it turns out it points pretty strongly towards one type of noun, and that is to protein. So a lot of origins of life thinking these days is about what's called an RNA world. What I want to convince you is to think about a proteins world. So this is the basic problem. Everybody here, I suspect, knows this part of the problem, the evolution bit. You came from your parents, they came from their parents. Our species came from earlier species. Descent with modification is what it's been called, or heritable variation. So we know about Darwinian evolution, this process, and in principle you can roll that tape all the way back to the very first cells 3.5 billion years ago. Now the question is, there's a singularity here in a sort of way, which is to say, I can roll that tape all the way back to understanding the first cell perhaps, but how did the first cell happen? Came from molecules. Something really different must have happened. And yet at the same time, something very similar is important. That is to say, this dynamics, we need to understand this dynamics if we're going to understand some of this dynamics. That's one of the points I want to make. Now here's a couple of the popular memes that are out there, or themes that are out there, if you like. The RNA world idea is that RNA came first. I'll say a few more words about that. The selfish gene is the idea that genes drive the whole program. They're the drivers of biology. The question is why, and that's the question that I would like to focus on. And then there's this blind watchmaker. There's a bunch of other books along these lines too. Blind watchmaker is the idea that something as sophisticated and complicated as living systems, how could they have come from just stochastic random processes? If I see a watch lying on the ground, I know it must have been made by a watchmaker, that sort of thing. What we need to figure out is how you get order from disorder. And I want to focus on the two kinds of molecules here. So on the one hand, as I say, you could be thinking about all kinds of biomolecules could have been the first. But I think that there's two that are crucially important for us to really focus on, and that is either the nucleic acids, RNA and DNA, or proteins. And the reason for that is because what we fundamentally have to answer, the real heart, the beating heart of biology, is sequence, structure, function relationships. And until we can get that, we're not going to have the origins of life figured out. So you can get metabolism, you can get chemicals going and that sort of thing, but the question is, how do we then start getting coding? And so I would argue we're looking for something like either RNA or DNA on the one hand, nucleic acids, or proteins. These are our two main options for codable function and codable information that specifies biology. So here are, I'm going to frame this as a sort of a whodunit. Like we're looking for a story here. That is, the question is, what's the origin, the mechanism? We're looking for a story, and this story will have answers that look something like a whodunit. That is, there's a who. In this case, was it RNA or proteins? There's a how. What is the physical process? How did evolution get started, and why does evolution keep running? There's a why. What are the forces, and towards what? A lot of thinking is, gee, if I could get something, a molecule to look like biology without having an enzyme or something, perhaps, then it would go towards biology. Well, why would it go towards biology? What's the driving force that would make chemistry turn into biology? And finally, who wins? That is, what is the fitness? Cells, of course, have fitness landscapes. Biology has fitness landscapes. What was fitness before, when there were just molecules, before there was biology? And why was there any fitness at all? So now I'm going to focus on proteins. This is the story I want to tell you. And so as you all know, proteins are the function molecules in biology, mostly. Certainly RNAs can do this too, some. Proteins take actions encoded by sequence. So I show here a simple model that we worked on many years ago called the HP model. H are the red beads, hydrophobic, and blue are the polar beads. They're the ones, so that it's basically, this is all nothing but oil-water physics. If you string together hydrophobic things that don't like water and polar things that do like water, and you string them together in specific sequences, what we learned many years ago was those things will fold up quite uniquely based on the sequence. Those unique things will determine the native structures of proteins. This is the main driving force for protein structure determination, and then those things will have molecular functions. This is my cartoon of sequence to structure to function, and it's mostly proteins that do this. RNAs, there are some that have function, but mostly RNAs are sequence to information. Now I want to focus on two parts to this problem, these two driving forces for us in trying to figure out origins. And the first of them is what I'll call needle in a haystack problems. Now the needle in a haystack problem, along the top I'm going to show you the needle in a haystack problem that all of us in the protein folding field over the last 40 years struggled with. 40 years ago, everybody said this is, one of these astronomical problems, and how do you find the needle in a haystack and so on. Well, the statistical mechanics community that we were in figured all this stuff out over the last 40 years. We now know how proteins fold. And what I want to point out here, and the thing that got us motivated to think about this problem in the first place, is that the origins of life problem has pretty much the same needle in a haystack problem. And that, to us, was a hint that this is where we should be looking. So first of all, let me tell you about the folding problem. What the folding problem was, it looks like this. I have a given protein of chain length n, it's got n amino acids in it, and I'm looking through its conformational space, that's the haystack here, to find the native structure, which is one conformation, that's the needle. I'm looking for the native structure out of a whole conformational space. Now, ballpark, and the arguments back then certainly were that I have this conformational quantity Z, that's how many rotational isomers do I have around a bond. It's of order 3-ish, plus or minus a bunch. And then I have n amino acids, then I would have 3 to the, so a typical protein is 100, 200, 500 residues long. So if I looked at, say, lysozyme, n is 130 amino acids, 3 to the 130, 10 to the 62 is the haystack size. I'm looking for the needle in that haystack. So I'm searching through 3 to the n to find the one conformation. The way this was framed back then, and has been often, is as a golf course landscape. That is, the search process is like I have some kind of a potential function, and the lower the potential function, the better. But the problem is that the entire haystack space is all flat, so nothing gives me any hints about where to find the golf course hole. And moreover, this is in this case, 130-dimensional space. So if you put in all the atomistic details, it's a much bigger space than that. So the basic idea was I have a golf course of 130 dimensions. It's all flat. How am I ever going to find the hole? And this is really how the needle in the haystack, how the protein folding problem was framed as needle in the haystack. Now here's the origins problem. It's not very different than that. First of all, it's also about proteins, but this is about sequence space, not conformational space. So what do I mean by sequence space? Well, here's my chain of n amino acids, n again is 130 for a lysozyme. But now what I'm looking for is how many sequences are there? Well, okay, at this position I've got 20 different amino acids. So now my sequence space size is 20 to the n, 20 to 130. So that's around 10 to 170 in this case. Again, blind watchmaker books and so forth all frame the problem as this sort of problem, that I have independent random steps and I'm looking for one state out of that. And it's very much framed in that way. But we know the answer for protein folding. And that tells us a whole lot about what we're looking for, I think, in the origins problem. There's 3 parts to the answer for the protein folding problem. The first is the landscapes are not golf courses, they are funnel shaped. And so the bottom line is, if you imagine just putting your golf ball on a funnel, it just rolls downhill and finds the answer. And that is a part of the explanation for why it is that real proteins tend to be able to fold in somewhere between microseconds and milliseconds. Proteins never had a folding problem. We had the folding problem. And so proteins do this very quickly because the energies keep bringing things downhill. And we know something about how that happens. It's about cooperativities. And so we also know the kinetics of this process. The way it works is it turns, this is work that Soren did and Bonnie Berger did, was to show that this HP model is NP complete. So in principle, if you want to find the global optimum for a given sequence, that can be very hard. And in principle, that can be very slow. But again, proteins work by physics and don't necessarily work by mathematical proofs. And so, but it turns out you can show through simulations on sort of toy protein data banks, if you will, that there's a strategy that can get you to the true global native almost every time, not every time, but almost every time by a very simple procedure, and that is local first, global later. Just make local decisions. Those are easy. You just search a little bit of conformational space. More global, more global, more global. And that's how we know that proteins fold up this way. They have a 4-helix bundle. You form this helix or that helix. It's all by random steps and so forth. But once you form helices, it's a little bit more stable than the thing that came before it. And that stabilizes and templates and so on. And so it's a procedure that goes from local to global. And so we know to look for things like this. First of all, we need to understand the cooperativities. In proteins, the cooperativities are because you have hydrogen bonds, you have hydrophobic interactions. One step at a time stabilizes the next thing, stabilizes the next thing, and each one reduces the entropy of the remaining space. And that's where the funnel comes from. So we know funnel landscapes give us the density of states. In StatMech, this is the density of states that we found and looks like this. This is the kinetics of the process. And the folding code is basically binary. We know it's hydrophobic and polar. We have, instead of the 20-letter code, what a protein is trying to deal with is its 2-letter code, its oil-water code. How do we know this for sure? We hypothesized this a long time ago, but then 2 beautiful experiments came along in science by Bob Sauer's group at MIT and Michael Hecht's group in Princeton, pretty much within the same year that we made the prediction, and what they showed was, yes, there is a reduction from 20 to the n to 2 to the n, that is 100, that is 10 to the n orders of magnitude. And that is to say, the main code is simply, I don't care if this is a leucine or a tryptophan, this residue, I care about whether it's red or blue, that's all. And if I get that bit right, I'm mostly there. And so that also is a huge reducer of the haystack. So number 1 problem that we got motivated by, needle in the haystack problem. Number 2 is the dynamics, the kinetics. How do I get evolution? This is really interesting. So this came up before. How do we define life, I think in Craig's talk. How do we define life? Now, and it's difficult, and there's been lots of shipwrecks on those rocks trying to define it. You know, if you say life is something that metabolizes and grows and duplicates and all that sort of thing, candle flames will work. So you need to be a little more careful than that. Lots of definitions people have gone through. But this is the NASA definition. It's something of a consensus definition, and it's the one we use because it has a feature in it that most of the good ones now have, which is really quirky, but it's interesting, and that's this. The NASA definition says life is a self-sustaining chemical system capable of Darwinian evolution. Now, I underlined the bottom part. I did that because there's two aspects of this that make it really funky. Aspect number 1 is you're defining a thing, not by the thing itself, but by how the thing changes. You're saying, I can tell you what life is if you can tell me how it's changing from one time to the next. That, to me, is a weird way to define anything. Second point, though, is it carries a really interesting implication, and this is kind of where we take off, and that is, if I can't tell you, in principle, if I'm trying to write down a model for origins of life, how did chemistry turn into biology? I can't tell you an answer to that until I can tell you how it's changing. So part of my definition of that transition has to be, how did we get Darwinian evolution? That is to say, life can't originate until it can propagate. So then we spent some time thinking about Darwinian evolution as a machine. that is to say, could we just write down, everybody knows how Darwinian evolution works more or less, depending on how far in the weeds you want to go with that. But the gist in our minds was, could we write this down in terms of the dynamics of the process, just capture the known dynamics of the process in a little bit of math. And our model looks like this. It says, so here's some time steps. Oops, that's not working. It says, here's time t, here's the next time step, and then here's the next cycle. These things are going through cycles. We call it a machine for two reasons. One is it's got to be plugged in. And by plugged in, I mean this is resource driven. So it keeps running because it eats. Food goes in, and so this thing keeps running throughout time, number one. And number two, because it's cyclically doing something. So we call it a machine, Darwinian evolution machine. So at time t, what do we have? We have a population of cells, call them X. At time t plus 1, we have a mutation. We call this the momsamble. This is an ensemble of all possible moms. Moms make moms. That's our basic scheme of things is moms make moms. They make daughters and moms and so forth. But the gist of the autocatalysis that's in Darwinian evolution is moms make moms, and that keeps running. So we call it a momsamble. This is the ensemble of all possible mutations from the mom that you have. And that mutation now grows up to become a population Y. So you now have a population X. These are the wild type. This is the mutant. You compete the two of them against each other. One of them wins, and then the winner gets more resources in the next cycle, and that's basically the machine. So it's moms making moms. They're autocatalytic sets. This is crucial for evolution because it would die if you had negative feedback. You need positive feedback. It selects on phenotypes, not genotypes. This is a hint that we're needing to look at function in proteins. Fitness is about differential growth, and most of the growth of a cell is duplicating proteins. Winners win resources, and this is really, it's an evolution machine. It's a search and select machine, and I'll say a word or two more in a minute about that. So we wanted to make a model. How might we think about a plausible start for prebiotics? And so, again, our focus is on proteins. I'll come back to the reason in a second, but we know that the Earth had amino acids from things like the Miller-Urey experiment. It goes back to the 1950s, and then all the current variants of it. We also know that peptides are makeable in simple prebiotic conditions. You can even find peptides on meteorites. We presume they happened on catalysts. We call it the founding rock, some little rock in Nebraska somewhere in a warm little pond, something or other. There's some kind of a catalyst that polymerizes amino acids. And we have to assert, any model has to assert some form of drive, has to assert that something out of equilibrium in the environment is driving a molecule-making process. Without that, I don't see any way to get to a living system. But here's the questions. All of this is fine, but it raises all the big questions. The big questions are, yeah, but how do you get long chains? This has been a crucial problem in prebiotic chemistry. You can make short chains, they're peptides, let's say. You can also make short nucleic acids. But the problem is they don't do anything. They don't grow, they don't encode anything. How do we get sequence function? How do we get autocatalysis? How do we get something that persists and adapts and so forth? And how do we get order from disorder? So what's required of any model, I'm going to show you our model in a second. There's several things that would constrain any model. One is you have to assert what is the non-equilibrium that's driving something. So there's day-night cycles, there's wet-dry cycles, there's things like that. But the environment today and 3.5 billion years ago had tons of things out of equilibrium. So there are lots of drivers, fluid flows, diffusion, a lot of kinds of things are out of equilibrium. The question is, how does that couple to some molecule-making process? Secondly, molecule-making process that's of interest here has got to be something about making sequence polymers, like RNA or proteins. It's got to start from randomness. I can't be the PI on a lab bench who invents the sequence and says, oh, look at this, it's a great sequence, it's going to duplicate itself. This is, if I'm going to create origins, I have to be thinking about how this came from randomness. And I have to get autocatalysis, and I have to get fitness to emerge from all this. So here's the problem, is that a lot of the prebiotic syntheses or something like this, you have the monomer units, you have some kind of a catalyst that polymerizes. You make mostly short chains. This is as a function of chain length. This is a log scale. We call this the Flory problem because almost all polymerizations have something that looks like this. You get an exponentially dying off distribution where you make mostly short chains and fewer longer chains. And so these ones are prebiotic syntheses of amino acids and nucleic acids, but you also find it in synthetic polymers. So the thing is, the point of this is just to show it's not so hard to make short things, it's particularly hard to make longer things. So that's the question of how do we get to something longer. But a lot of these polymer problems are all focused on homopolymers, meaning I either make oil-like polymers in oil-like solvents, like this is how you make carpets, for example, or I make water-like polymers in water-like solvents. But what we have here for amino acids is HP polymers, that is, they're partly water-like and partly oil-like, and that's the magic of them, we would argue. So what happens is, okay, now I have hydrophobic and polar residues. I will get a bunch that don't do anything useful at all. They can't fold up. For example, the all polar is just a random coil in water. The all hydrophobic just aggregates with other chains. So you have to be somewhere in the middle, but there's a Gaussian distribution because in the PDB you have about half hydrophobes and half polars. So there will be lots and lots of sequences, just random sequences, that are pretty good folders. And that means they're down here somewhere. They either collapse into like molten globule states, which are not quite folded, they're sort of smeared around a little bit, but they're compact, or they actually fold into something native-like. Now, prebiotically, probably not many of them did that. And then a fraction of these, of the folders, are what we call fold cats. That is, they can fold and catalyze. Now, it's not weird to think about proteins as catalysts, because that's what they do for a living. But it might be where your imagination gets stretched a little bit is to think about some random chain sequence folding up and having a surface. Now, the surface here, in this case, just for model purposes, we use as simply a set of hydrophobes. If there's a few hydrophobes, I have a process that can look like this. So here's a chain sequence. It's got enough hydrophobes that in water it collapses. It's got a hydrophobic core, but it's also got a few hydrophobes on the surface. Most proteins do. A few hydrophobes on the surface, what that could do is I have another chain that's got two hydrophobes. It sticks to the catalyst chain. Another monomer comes in. By the localization, that basically reduces the kinetic barrier. Menger has written an article about looking at that mechanism in general in today's proteins, and the acceleration factors can be quite large. Probably early on they were not large at all. So here's what the mechanism does. The mechanism takes this Flory distribution, which says I'm not going to get very many long chains, and what it does is it extends out this distribution. That is to say, it creates longer chains. Now, why does it do that? There's two factors. If you're making HP chains, what happens is even the short ones will collapse. It's just oil water. They'll collapse. And when they collapse, they protect the hydrophobic core from being hydrolyzed. Water can't get in, water can't destroy the chain, so it has a slow degradation process. Second is that chain has an ability to catalyze itself, the elongation of other chains. So those two factors give you this. They also give you these autocatalytic sets where certain sequences can help you make other sequences. And now you start getting sequence structure function relationships. And this is showing, this is sort of schematically of that Flory plot. Green is the Flory curve, the typical Flory curve. Blue is now what happens if you look at this. We do computer simulations of this model. And what you find is these are short, this is sort of concentration of short chains. This is concentration of intermediate length ones and longer ones that are fold cats. And what happens is first of all, once chains fold, they degrade slower, so they persist longer. And secondly, once you have ones that are able to catalyze, they can help these other ones get longer and longer. So that's basically the argument. There's these two forms of cooperativity. Now, as I mentioned before, did I mention this? No, let's see. Okay, somewhere way back here, yeah, okay. Back here, I mentioned one thing that's crucial is you need to get some form of cooperativity. This is missing on this slide. I thought I had it on there. But when you invoking any model for origins from chemistry to biology, you have to invoke some form of cooperativity. That's the essence of what we're looking for. The problem is often framed in terms of, gee, the probabilities are too small. Well, that's not really the problem at all. I don't care what the probabilities are. This is true for actually lots of StatMech and most disordered order transition in statistical mechanics. And snowballing, if I take a snowflake and ultimately get a snowball and an avalanche, it was just one snowflake that started it. The problem isn't what the starting probability of something is. The fact that it's infinitesimal is not the issue. The fact is, how do I take something that's infinitesimal and take it to the next stage of being slightly less infinitesimal? If I can assert a cooperativity factor, then I can get from small probabilities to larger ones. Okay, oops. And so that's what we have asserted, these two factors. These are factors that work in today's proteins. It's the fundamental physics of how proteins function. One is when a protein folds, it has a protected core. And the other is when a protein folds, some proteins that fold will catalyze reactions of various types, including elongation. So just a few more points here and then I'm done. And this is showing you that the Foldcat process leads to, this Foldcat process leads to a funnel in sequence space. This is not the conformational funnel of folding. This is not in conformational space of a single chain. This is in the space of the whole sequence of all possible chains, not only of a given length, but of all lengths. And what it's showing here is that I start off with little peptides, every conceivable little peptide. This is totally random. This thing is a junk generator. That's all. It's just making little peptides. Almost all of that junk is just junk. It doesn't do anything. But some of them are a little longer, and they get elongated by the ones that are even longer. And so you get a funnel. We can compute very simply what the slope of the shape of that funnel is. It turns out to be a straight line on a log plot for simple reasons, but I won't take your time on this. Bottom line is, it's a funnel in sequence space. And this is the basis for the disordered order. So there's no PI designing sequences here. This is really a complete, starting from complete junk, and just an incredibly small fraction of junk percolates, and that's because of these cooperativities. And to contrast that to the other view, so this, I'm calling it the Foldcat process, generate a lot of junk, but it's selected by competition and fitness. This is Darwinian. The Darwinian process is giving you the answer here. The Darwinian process is very simple. These things are just competing for resources. They hydrolyze, so they turn over. And so if I'm a crappy little short thing, I will get degraded and my amino acids will go back into the soup and the longer ones can pick them up. And so that's the process. It's an evolution process. This is not a self-copying process like an RNA world view might tell you. It's not about self-replication where I start with a clever sequence and I just make copies of it. The dynamics has features that look very much like folding dynamics. This is the dynamics from evolution, from the Foldcat hypothesis. First, what happens is you make tons of short chains. This is, by the way, both of these are log scales. I can't tell you anything about real time here. This is a computer model. And so I'm guessing, this could be 100 years, it could be 1 billion years. I really don't know. And this one is a log scale too. But here's what happens. At first, what you get is just a lot of peptides, junk peptides, lots of them. But after a while, what happens is you start getting ones that are longer, and then they become Foldcats. And so this is the process. It's sort of short to longer. This is the sequence of events, and it resembles that figure I showed a few minutes ago of a sort of a conformational space kinetics, which is local first, global later. This is short first, longer later. What we know is, so every model has parameters in it. This has 3 parameters in it, and they have to do with the amount of cooperativity to give you the dynamics. And the key point here isn't what these parameters are. The key point is that there exist parameters in which you can leap from chemistry to biology. This is a terrible region to be in. This is prebiotic chemistry. Everything just keeps falling apart. And that's where most everything is to begin. This is a wonderful world where we are now. This is where biology is, where everything keeps rolling. Evolution keeps rolling. But it turns out there is this region right here which is very much like any other phase transition. Think about the folding transition. I start with a denatured ensemble, I change the temperature, or I change the denature, and I tip the whole thing to a point now where the parameters of the model, the sticking energies and all that stuff, are sufficient to now drive everything over to the native state. I have now gotten over that kinetic barrier. And that's what this state is up here. When I get to certain values of these 2 key parameters, I now go over that transition. This is the tipping point. One last point, transformative for the origins of life was when catalysts came to be mobile and mutable. To me, this is a really interesting point that doesn't get discussed much, and that is, okay, I got a founding rock somewhere in Nebraska. Okay, maybe I've got life cooking there. Something's going on super interesting. But now what? It's all stuck in Nebraska. Nobody wants that. And so ultimately what you have to get to is something where your catalysts are small enough to be able to go into cells in an individual way, are programmable enough that you can change the catalyst to be able to make it work under different conditions, and are mobile. It can float away from that little pond or from the founding rock. Now, I like this metaphor that there's an aspect of the Industrial Revolution that sort of illustrates this point. And that is when Industrial Revolution, energy was no longer tethered to rivers and waterfalls. You know, back in the day, you were making material or wheat or something like that. You needed to be near where flowing water was. Industrial Revolution gave us motors where you carried your energy on board, so steam engines, internal combustion engines, things like that. That opened things up in an unbelievable way when you could mobilize energy's utilization. Same thing with iPhones. You know, I'm old enough to remember when phones were hung on the wall like that picture that we saw this morning. And now we have iPhones. That also is transformative for society. For biology, in the origins process, what's transformative is when catalysts were no longer this big macro rock, but they became microscopic. And you need programmability. Why? Because think about a cell. In a biochemical pathway, everybody's got to run at room temperature or biological temperature, and has to be able to work with the same substrates and so on. There's a lot of tailoring in building a biochemical pathway that can produce something under all the conditions, identical conditions for every step. All right, final point or two here is so I'm always asked about, okay, why couldn't that have been RNA though? Because RNA can do a lot of the same things proteins can do, some of them can. But there's pretty good reasons for thinking that it's not general enough. RNA is simply not generalizable enough to have done the things we care about. First of all, evolution acts on phenotypes, not genotypes, which means actions and functions and not memory. Actions and functions in RNAs are pretty limited. And where the function comes from, the difference in physics is really interesting because you get function from the compactness of structures, that is to say, folding in a protein. And the problem with RNAs is they're too stringy. So the stringiness gives you a couple of problems. One is you have a very bumpy landscape. RNA landscapes have these huge barriers, and it's because you have 3 hydrogen bonds per base pair. Those are very strong, so you get very deep wells, and barriers are pull this one apart, shift it up to here, shift my secondary structures. You get very stringy structures, and you also get ones where you can't code them very well. Encoding means I take a sequence, I know what the answer is I'm going to get. I change the sequence, I'm going to get a different answer. That's what you get in proteins. One sequence gives you one fold. The mapping is very different in RNA, though. Lots of one sequence will give you lots of different folds. Even one sequence, I think this was Christine Guthrie had a sequence once that had like 7 folds in it. And anyway, just to end up here, I don't think I've told you the whole story. I don't know the whole story. The story that we've tried to paint here is about how do I get to a stable evolution world where evolution can then make its own choices about what the molecules should be. And so I'm drawing this in some grand potential diagram. And so what we get to here is this. You have a stable world of folded and catalytic peptides. It's self-sustaining, it keeps running, it evolves, it starts choosing better things and so forth. Then the later stage, now all kinds of the really powerful, interesting stuff happens. You get heritability, the genetic code, RNA and DNA, you get cells and lipids and that sort of thing, biochemical networks. You don't need any of this stuff at the first stage to be stable, but you do need it, of course, before you define life. So the way I would define life, you don't see it until you're out here. However, the NASA definition of life, interestingly, is actually only this. I just need Darwinian evolution. So, summarizing our whodunit story, it's the proteins. What seeds it all? It's just some persistent maker of short peptides. The positive feedback comes from folding and catalysis. The action is a sample, compete, and select. It's a Darwinian action. It's not a copying action. I'm not looking for self-replicators. I'm looking for something that can take random junk and then compete it. The physics is just oil-water physics and polymers, and the goal was not biology. The goal is just persistence. It's just this little fold happens to live a little bit longer in the environment than that one did, and the environment keeps trying your patience on this, and so this one lasts for a little while, change the environment, now this one lasts for a little while, and so the environment pumps us along as we ultimately get to more persistence. And finally, these are the people who did the work. Thanks to them, and happy birthday, Soren.
I should have emphasized this is a distinguished John von Neumann lecture, part of that series. I don't think I said that before. Any questions for Professor Dell? Yeah, Craig, please.
Just a couple of observations. You're trying to solve pretty much an impossible problem. We know the end answer. We know the results of protein folding. We know the results of all life forms. You can't get a living cell without nucleic acids as a template for proteins. So you take out the DNA or even RNA viruses, you can't have any life or reproduction. So it doesn't mean the origins had to copy that in any way, but also built into the protein folding, as you know, it's not perfect. And we have mechanisms in our cells, virtually in all cells, to remove the proteins that didn't fold properly. Right. We have built into the coding the longevity of a protein, whether it's seconds, minutes, or hours, but usually not longer than hours in most cases. So very quickly, if it folds wrong, it's eliminated. Even if it folds right, it's eliminated. And I don't know how you and Soren built into your models the chaperone problem. So lots of things to throw out at once.
Okay, thanks. Yeah, lots of good, interesting stuff. So first of all, on the chaperones, we didn't build it in, but there's lots of chaperones that are pretty simple-minded. They're just little hydrophobic surfaces. So today's fancy chaperones are more complicated. You get these barrels of, Romiel, for example, and stuff. But let me go back, because this is sort of your key point. I take as your key point, and I agree with you 100%. So this is the place where I think we got to so far in our modeling. This is the place you're looking for, we're all looking for. This is the origins of life, and we're still here. And so Right, no DNA, no cells, no biochemical networks, no nothing. But here's the thing. So there's a couple of points here. One is, my little pond in Nebraska can be perfectly stable because I don't have predators yet. So this thing can be very happy. And then once I get out here, now I'm going to have predators and everything else, and I got to compete at a much higher level. The other advantage, the big advantage as I see it, and sort of the reason that RNA and DNA and cells, all that stuff, all the rest of biology got recruited in the first place, is because if you think about even sticking with persistence as a measure of fitness, that's a very simple thing. It's a physical thing. It's not the only thing in biology. There's a ton of things. But even if you just stick with persistence, what RNA and DNA and cells do for you is they extend the persistence almost to infinity, because the thing is, when I'm here, I'm just a single molecule and I will always decay. As you say, proteins will turn over even if they're folded well and all that sort of thing. Molecules, individual molecules just keep turning over. But if I have a lineage, this is a beautiful way to extend persistence essentially out to infinity. So if I'm just proteins for now, and I'm trying to figure out, okay, how do I evolve any further than that? The thing I most want to do is encapsulate into cells, and I want to get RNA and DNA involved. And now why would they happen? So we are working on trying to get to a genetic code. Why would you recruit RNA and DNA? Or how would you recruit it? So the why is the persistence is much larger. The how, it turns out in our level of modeling is very simple, and that is, if I'm a protein and I can bind to little nucleic acid dimers, trimers, tetramers, things like that, it stabilizes the fold. And anything I can do to stabilize gives me more persistence. The more persistence I can get from whatever I can do, folding is all we did so far, but if I can stabilize, increase the persistence also by a binding event, that's a good plan for how to get to the next level of persistence. And so that would then tend to generate ways for RNA and DNA to come into the picture. There is a lot of work that people have done on looking at nucleic acid, little peptide interactions with little nucleic acid trimers and so on, in different kinds of solvents. And there is probably something like a physical code there. I think it's got some hydrophobicity in it and charge in it and that sort of thing. But anyway, I think there are reasons why that recruitment could start to happen.
I think we'll take the rest of the questions offline because the food's starting to get cold.
Yes.
Thanks again.